Good morning. Can everyone hear me? Jenny, can you let me know if I'm being heard?
Yes, David, we can hear you.
Okay, great. Um,
Do you need to
well,
turn your camera on?
oh, I guess I can. I don't know. You're asking a lot of me. Yep. Okay, good, great. Um, I'm David Schwartz, Chair of Medicine. I want to welcome all of you to our annual Research Day. For me, it's one of the most exciting days of the year, and it's exciting because we get to celebrate each other's accomplishments in terms of creating new knowledge. And that for me is a particularly rewarding time of the year and a rewarding event in terms of recognizing uh, not only how substantial these accomplishments are, but the spectrum of the research accomplishments from very basic research to translational research to applied areas of investigation. We have a number of our faculty today presenting at Research Day, and I do look forward to those presentations, as many as I'll be able to attend. And we also have a guest from the Brigham and Women's Hospital, Julie Silver. And Dr. Silver is going to be speaking about diversity and inclusion, taking a data-based approach. And I think that that is particularly timely and important to the strength and the breadth, not only of our research operation, but our clinical and educational programs. I do want to say that we've made quite a bit of headway in terms of our educational programs. 10 years ago, when I began as chair of medicine, we had about 3 or 4% underrepresented minorities in our program. Now we have consistently between 15 and 20% underrepresented minorities in each one of the intern classes. And this year we had 19% of the class made up of underrepresented minorities. This actually has resulted in an improvement of the quality of our house staff. Again, when I started, about 5 to 10% were in Alpha Omega Alpha, the honorary society for medical students. Now we consistently have about 40% of our house staff in Alpha Omega Alpha, and this year we had 38% in Alpha Omega Alpha. We also this year were on average about 100 less in terms of the extent that we had to go up in the match to constitute our class. So we got a much higher caliber of medical students applying to the program and also being selected for the program, selecting into our program. So, creating an environment of underrepresented minorities throughout our medical center and throughout our training, education, research, and clinical programs will only strengthen our faculty and strengthen our trainees. Back to the research, I want to thank our presenters. I want to thank the organizers for this conference, and I most of all look forward to the science that's going to be presented. I know this is a trying time for all of us, but from my perspective, the way we're going to get through this is to understand this disease at a very basic level and also at an epidemiologic level. And if we can understand coronavirus better, both in terms of the epidemiology, basic pathophysiology, as well as the treatment options, we're all going to get through this together. So let's hang in there, have as positive an attitude as possible, and work together to address this crisis that's in front of us. I look forward to the talks, and I'm going to turn it over to is it Tanya Reese is going to be the first speaker, or how are we going to deal with introductions? Jenny, help me out here.
Hi, so Christy Kuhn is going to give our first introduction.
Okay, great.
Okay, so I would like to share this with everyone. So to start, I would like to announce this year's award in mentoring. The Department of Medicine Mentoring Award this year goes to, Dr. Arthur Gutierrez Hartman. He is a professor in medicine in the Division of Endocrinology in the Department of Medicine and the Associate Dean for Research Education. In addition to having a highly successful research career over his 35 years here at CU, he has mentored hundreds of successful individuals directly and on mentoring committees, including some of the outstanding career scholars as well as faculty leadership. I can personally attest that his mentoring of individuals is lifelong after he serves in the initial formal role. He developed the current medical scientist training program, or the MD-PhD program here on campus, as its director for the past 35 years, obtaining and maintaining its NIH funding and vastly increasing the diversity of the program. He is the founding director of the Physician Scientist Training Program track within the Department of Medicine Residency Training Program, which promotes the development of physicians as scientists during their postgraduate training. Both locally and nationally, Arthur serves on numerous advisory committees for training and diversity. And because of his passion for mentoring and training a diverse workforce of physicians and scientists, I am honored to present Arthur with this year's Department of Medicine Mentoring Award. And so, Arthur, if you want to unmute, I'd happily allow you some time to share any thoughts with us.
Thank you very much, Christy, and I'm really honored to receive this award, and particularly in name of Dr. David Schwartz, who's really been a luminary in the Department of Medicine and really taken the department to a significant next level. That's just amazing. And it's been an honor to be a faculty with Dr. Ridgway, who was here and recruited me, and with Dr. Schwartz. And I just want to say that it's really an honor to have mentored as many individuals as I have. And I don't really want to take much more time. I've been asked to introduce Tanya Rice. And so, Christy, if that's okay, I can go ahead and do that.
Yes, please go ahead.
So can I just say, can I interrupt let me just take chair's prerogative, excuse me. I have had the honor of serving on a thesis committee with Arthur. And it is one of the most enjoyable experiences. Arthur not only has a great relationship with all of his MSTP students, but he is able to understand the science outside his field in a way that allows him to bring value to the discussion and think about other ideas and bring up other perspectives that individuals very close to the research sometimes don't see. He does it in a way that makes the students think rather than fear his comments. And I think that's the great sign of, that's the sign of a great mentor, helping their students in a very Socratic way think through a problem and extend their concepts to areas that they hadn't conceived of before. So Arthur, congratulations, well deserved, and I'm really pleased that you received this award.
Thank you, David. Thank you very much. Our first speaker is Tanya Rice, who comes from one of my favorite countries, from Portugal, and she's taken an unbiased genetic approach to really try to understand the genetics of homeostasis. And in doing so, she's used Drosophila as her model system, and has taken a really clever approach that she will tell you about. And she's going to give a talk on how flies get fat, from genes to neurons. And it's been my pleasure to also have helped mentor Tanya along the way. And so I'm really excited to hear her talk. I'm going to go ahead and unmute and stop my video. And again, thank you very much to the committee and to Dr. Schwartz and Christy Kuhn and others. Tanya, take it away.
Um, Thank you so much for the opportunity to present my work. And as Arthur said, it's a few doors down my office, and I run in with several different questions. I came to CU a few years ago as an outside hire, and Arthur has helped me through my science, but also invaluable mentoring through understanding and navigating the system. Especially being a basic researcher on a clinical department, which I have been enjoying quite a bit. So I also, full disclosure, I am at home. I have two younger kids. Everyone is being instructed to be really quiet and stay away while mommy is at talk. Sometimes things don't go that way, but I'm hoping that they will. Okay, here we go. Thank you so much for the opportunity. And again, thank you, Jennifer, for allowing us to, independently of the circumstances, being able to continue to share our science. And I'm going to, are you seeing my screen now or not yet? Not yet, okay. Okay, so here we go. So since this work started in Ishar Hariharan's lab at UC Berkeley, and my biggest question was not necessarily trying to get into mechanisms of obesity and energy homeostasis, but mostly trying to understand the plasticity that exists in biology within an organism. You know, we see a lot of times phenotypes can be quite plastic, sometimes quite resilient, and when things break down, we get disease. And I chose to study energy homeostasis mostly because there seems quite a bit of challenges, you know, for individuals within the population, but also single individuals throughout the day. Your metabolism changes greatly throughout your lifestyle and lifespan. So it's a very complex question. All my background was in fruit flies, studying fruit flies as a genetic model system for disease. I came from a cell cycle and growth control background, and I really wanted to adapt flies to develop a mechanism to study these questions. So when I started, there was not much done in this question. And again, we know that in our department and in our division, we know that there's a lot that goes into obesity. We don't refer to it anymore as a behavior issue, but a true disease. We know that there are a lot of people that are obese, and definitely the current lifestyle with increased sedentary behaviors and higher availability of foods have contributed to the development of the disease. But despite these, we know that there are several individuals that are able to maintain less states of less increased fat. On the other hand, there are also individuals that have what we call the healthier lifestyles with better diets and exercise, and still have challenges and maintain obese states. So we know that there's this clear genetic predisposition for obesity. It's predicted that about 60 to 70% of obese patients are so due to genetic inheritance. And it's also predicted that we maybe only know about 5% of these genetic predispositions of obesity. So this is where I started, and I decided to use fruit flies to model to study and model this issue. So, and why flies? So basically, flies are just really simplified little humans. So there's clear genetic counterparts to much of our genes and pathways, yet we don't have in flies the elaborate gene families that we have in humans. Usually have a single representative of each gene family. So if you're thinking that you're trying to uncover a known genetic basis for obesity, you definitely want to do it in a more simplified way. Very important, they have all of the orthologous organs that are required for energy balance. So they do have a complex central nervous system that regulates food intake, food preferences. If you put flies in a choice food assay where you give them a bitter versus a sweet food, they will prefer the sweet food and they'll eat more of that food, and they do get fat on that food. So they have a central nervous system that regulates complex behaviors like food choice, activity, sleep, all that. Unbiased genetic screening and all kinds of different molecular biology and behavior tools have been well developed in flies, and we can do tissue specific or even cell specific genetic downregulation or overexpression of specific factors to study obesity. Most importantly, they also have an organ that is dedicated to energy storage, which is called the fat body. And there's a little bit of a difference between mammals. The fat body is mostly a fatty liver, so it stores both triacylglycerides and glycogen. Flies also regulate their levels of circulating fats and sugars in the same kinds of ways that humans do. So they have an insulin and glucagon ortholog systems. So I'm focusing on the larval stage, and this is because in the larval stage of fly development, most of what larvae do is eat and move to find food and store energy, and not much more. So therefore, activity, food intake, and energy storage and usage are very tightly coupled. So one of the biggest challenges to develop flies to study energy homeostasis and obesity was finding a phenotype. And this is because flies are insects, they have exoskeleton, and so they're very capable of storing more or less of their fat within the same size body. So just looking at a fly, you can't really say it's fat or lean. You can say that it's smaller or bigger, but that's size, not fat matter. So I had to develop an assay that could be used widely and easily and in a reproducible manner to try to do a screen to identify those not yet known causes of obesity. And this is where I decided to try density. So we all know that fatty tissues are really low density. And here I'm showing you two different genetic backgrounds, a wild type. And this was, to date, when I started, the only known fat mutant in flies. What I hope you can appreciate is that they're submerged in the same solution of PBS and sucrose. And you can see that most of these wild type larvae are sinking, and all of these adipose fat mutants are floating. So this was really exciting, and this is based on Archimedes' principle, and suggested that we could identify differences in fat in larvae by just looking at differences in density. So I applied this assay to do two different screens that I'm showing you here. One was a genetic screen. Both of these were small pilot screens. And in my genetic screen, I screened for a collection of 900 homozygous viable mutants to identify which ones would give me these differences in density. So which ones are floating, whereas most of the controls and the rest of population are sinking. So these will be our fat mutants. At this time, because floating could be, larvae could be just trapping air and not really having extra fat, what I did I developed gas chromatography mass spectrometry to measure quantitatively and qualitatively the differences in fat for these larvae. And I will not be showing you today, but what I saw was a positive correlation with the fatter larvae are, the better they float. So we definitely can use these difference in density to isolate fat mutants. And conversely, we can also use them to identify lean mutants. I focus on fat. So when I look at these genetic mutants that increase fat, there was a big amount of these that had genes that were had roles in the brain and they were neuronal genes. So at the same time, I decided to do a screen that would target more specifically, not genes, but areas of the brain to identify which areas of the fly brain regulate fat. So here I used a little genetic trick where I used a collection of 650 different drivers to turn on a dominant negative form of dynamin. Where these drivers are on, those areas of the brain are expressing this dominant negative form of dynamin. Neurons are viable and alive, but they're unable to recycle their synaptic vesicles, therefore they're not having synaptic transmission. And then I did my assay for these different crosses and identify 72 regions of the brain that gave these floating increased fat phenotypes, suggesting that it was necessary for those regions of the brain to be properly firing in order to regulate body fat. So 72 is a lot, and we didn't know exactly what regions they were. So I crossed those same drivers with GFP just to look and identify the areas of the fly brain that were responsible for the phenotypes. And I'm showing you here. So this is a schematic of a fly brain. There's 2 lobes, and this is a ventral nerve cord. And you can see here on the left of the screen, there's this area in green. And this area is a neurosecretory region of the fly brain. So if we silence neurosecretion in the fly brain, these flies come fat. So we're now focusing in a lot of these areas. From the 72, when we crossed them to GFP, we narrowed down to 16 areas of the brain that had overlapping regions of expression. And today I'm talking to you about this upper right area called the mushroom body, which is the area that is being shown since the 1960s in the fly to be required for memory and learning. So a lot of our drivers overlap in this region known as mushroom body, which is the region for memory and learning. And some of you might know that there's this idea in the clinical world that obesity may be a predisposition for early cognitive decline. But is it really? So what we're seeing here is that we are disrupting memory and learning and having an increased fat phenotype. So we're trying to discern a little bit these association. Is it really a cause or just an association? So to do so, we got hand into some more mushroom body drivers. So mushroom body, as you can see here, is collection of a bundle of neurons that organize into different lobes. In the field of memory and learning, the different lobes have been associated with regulating short-term, long-term memory, and also parts of learning. So we got first one driver that expressed in the whole mushroom body. And if we silence the whole mushroom body, I'm showing you here a graph that is looking at the density. Black here is the control, and blue and red are my experiments. You can see that, do I have some questions? Okay, sorry, I got distracted with the chat. Okay, so what we can see here is that the blue line, it's when we're silencing the mushroom body, flies start flowing faster, suggesting a fat phenotype. What was interesting to see is if we did the opposite. So I told you that dominant negative dynamin silences synaptic transmission. If we overexpress an activated sodium channel, we're doing the opposite. We're activating synaptic activity. We see the same thing. Flies are also fat. So this is suggesting that this bundle of neurons, it's a little bit more complex, may actually function as a buffering mechanism for energy homeostasis and has pro and anti-obesity neurons. So then we went to other lines, and I'm showing you here when we're silencing with this line that only expresses in gamma and alpha beta lobes. And we can see that then flies are super fat, really, really fat. This is the fattest we've seen. But if we activate it, then we don't have a phenotype. So now, through a collection of 80 different drivers, we're trying to pinpoint which regions of the mushroom body are required for anti and pro-obesity neurons. So these are neurons. We're manipulating neurons, but we're seeing fat phenotypes. that are the phenotypes are being observed in the fat storage cells. So there needs to be communication between the brain to the fat storage tissues to be able to change these levels of organismal fat. So what are the molecular players? So then what I did next, I focused on what is known about mechanisms of memory and learning in flies. And so the molecular mechanism in the mushroom body for flies has been dissected to how this neuropeptide called amnesiac regulates PKA signaling to regulate expression of the transcription factor CREB that will induce either synaptic strengthening or establish new synapses that are required for reinforcing memory or establish new learning. So since the 60s and mostly through the Seymour Benzer lab at Caltech, a lot of these mutants in these pathways are available. So what we did next, we grabbed the mutants in the pathway, so DUNS, and DUNS is here, which is the cyclic AMP phosphoesterase, and rutabaga, flies always have really funny names, does the opposite, and also amnesiac, and we test their metabolic phenotypes. What I hope you can appreciate in this graph here is when we have DUNS mutants, and so basically when we have DUNS mutants, we have increased PKA activity, these flies are really fat. When we have amnesiac, so the neuropeptide and rutabaga mutants, we don't have a fat phenotype. So this would do the opposite. When we don't have amnesiac or rutabaga, we have low PKA signaling. But what we observed was that despite not having a fat phenotype for amnesiac and rutabaga, what I'm showing you on this top graph now is levels of physical activity. So how much flies move around and exercise, how many calories are they using? We can see that both rutabaga and amnesiac seem to be moving less. So presumably they are in a positive calorie output, but they are not fat. And if we look here in this bottom graph, and this is how much flies are eating, amnesiac are eating more. So they're eating more, moving less, but they seem to be resistant to obesity, which is really interesting. And we're trying to discern where this coupling between food intake, physical activity, and overall organismal fat is. So what I'm showing you here is data for whole organismal mutants, and where we're trying to test is that it's within the mushroom body, PKA signaling is required for this regulation. So we did exactly that. We used the same driver that I showed you 2 slides ago that was necessary to regulate body fat in the mushroom body. And in those neurons now, I'm showing you here in red, we overexpress PKA. So we have now high levels of PKA only in those neurons. And you can see that these genetic background, this overexpression compared to the GFP control induces higher fat, and phenocopies what we observed for the DUNS mutants. So indeed, higher levels of PKA just in the mushroom body, the center of memory and learning, is able to misregulate organismal fat. If we do the opposite experiment, and now we have a dominant negative form of PKA, both heterozygous and transets dominant negatives, we see that these animals here in black compared to the genetic background controls are lean. So this suggests that indeed PKA and in the mushroom body is able to, as shown before, regulate memory and learning, but there must be some kind of coupling mechanism where it's also regulating energy homeostasis. And I'll talk a little bit more in the end why we think this is happening. But is there a coupling factor that will coordinate the learning and memory with energy availability? So what we had a little hint of what this could be, because in the genetic mutant screen now, we had a handful of mutants that were fat that had been implicated previously in memory and learning, that had roles in memory and learning. And I'm going to show you one of those. ARC is an immediate early gene. In mammals, it's been shown to be involved in mechanisms of memory and learning. And what we saw in our flies, and we published this, was the first paper in the lab, was that ARC mutants, as you can see here in red, have higher float faster, so have lower density and are fat. And we confirmed this with GCMS. If we did the converse, overexpress ARC in ARC neurons only, they are lean. So I told you that ARC has been known to be a learning and memory gene. There's even a more interesting aspect now that came out maybe a couple of years ago, where ARC has viral-like GAG domains and forms viral capsids that transfer RNA between neurons and muscles. And this is one of the mechanisms that we're now trying to look into, but I'm not talking about it today. But so ARC is an immediate early gene, and in flies it's been shown to be that, but it hadn't been shown to have learning and memory roles in flies. So what was important for ARC is that despite regulating memory and learning behaviors, it was fat not because they ate more or moved less. They actually ate the same and moved the same as the genetic backgrounds, but they did have a completely shifted metabolism. I'm showing you here just a summary of the metabolomics of ARC compared to genetic backgrounds into storage of fat and slow usage. So when we look at where ARC cells express, we saw that they express in the same area as the mushroom body and that driver that I showed you. So ARC seems to be expressed in the learning and memory center of the flies. The other thing that was really interesting is that ARC levels, the protein itself, seems to be regulated by diet. So here in this panel to the left, I'm showing you, these are now wild-type flies that were challenged with a high-calorie diet in darker blue to a low-calorie diet in lighter blue. And what you can see is that the lower the calories in the diet, the more cells in the brain will express ARC. So could ARC now be our missing link? So we know that it's in the right place, so in the memory and learning center of the flies, and it seems to be responding to calories. So as I told you, we didn't have a role for memory and learning in flies, so I tested that exactly. So here I'm showing you an associate of learning results that I tested wild type of the genetic background and ARC mutants. So what I did, this is a odor association where I paired odors with a sweet reward, and I taught flies through 7 sessions to prefer the sweet. So they always they associated the reward with the odor. When you test wild type flies, I also did the reverse turning. When you test wild type flies in the absence of the reward, you see that after 7 sessions of training, wild types will prefer the odor that was paired with the reward. When we tested these in the ARC mutants, they don't seem to learn it as well. So then we know that in mammals, and in mice specifically, there's a correlation between increased BMI and high fat diets with lower learning scores in different assays. So we did the same for flies now. These are now wild type flies where we fed them either a normal diet or a high fat diet, did the same associated assay. And you can see that flies in the normal diet seem to learn as well as the wild type flies. But if we give them a high fat diet, which we know makes them fat, they don't seem to learn as well. So we have recreated here the situation where flies that are obese don't seem to be learning as well. Remember that I showed you that in a high calorie diet, ARC is downregulated and ARC mutants don't learn as well. So this is now what we're testing. We think that ARC is our coupling factor that is downstream of PKA activity that will regulate learning and memory, and at the same time regulate release of energy that is needed for learning and memory. So what we're thinking of the why this model is in place, it's because the brain, in order to execute its function of learning and remembering, needs energy. So to do so, that should be important to couple that activity with the energy that it's needed to sustain it. So this way, when new synapses need to be strengthened or established, ARC would be coupling that with lipolysis that would induce release of different energy that would then come to the brain to sustain that growth. And indeed, it's been shown in adult flies that through the active process of learning, the mushroom body incorporates more glucose. So there's some data that suggests that. So now we're testing if indeed, for instance, higher levels of ARC, when you feed flies a high-fat diet, are able to rescue those defects in memory and learning. So this is a project that it's still in its pilot phase. It's not yet funded. I hope to get it funded soon, but we have all the tools and exciting preliminary data to test a lot of different hypotheses. So finally, I would like to just thank everyone that does the work. I have a very small lab. I have amazing collaborations here in campus, and I'm always rewarded with the best students for some reason. So my students and trainees that are doing the work, I do a lot of these. This is my pet project, so I do a lot of the I do a lot of the work on this project. And I also like to thank all the sources of funding, and also the department for I have had bridge funding that was very needed to sustain the lab for the past 8 years, and I'll be happy to take any questions.
Thanks, Tanya. That was a great talk. I'm going to encourage all attendees that if you do have questions, please post the questions in the chat function. So let's see if any pops up. So one of the questions I have, Tanya, is, it looks really interesting how these neurological functions are feeding back and integrating with energy metabolism and usage. How do you envision kind of, I guess this is going to allow you to wildly speculate, but that's something we all like to do anyway. How do you envision this is going to link back to the way we deal with obesity in patients? And you're still on mute.
Just noticed that. Thank you. So I think that's a very interesting question. So one of the things that's been exciting and at the same time overwhelming through the process of these screens that we've been doing is that realizing the diverse phenotypes, the diverse obese phenotypes that we find. So one thing to keep in mind is that some of these may be strictly autonomous on energy metabolism itself and not behavior at all. Others may involve the brain. Some are completely due to increased feeding, disrupted feeding satieties and others activity. And so that's one thing to keep in mind. So partly answering your question, some of them will require interventions maybe at the neuronal fat storage interface. Others will be just not involving neuronal at all. Some diets, manipulation of specific diets may be helpful sometimes, other times not. We have a mutant that the work is just coming out this month where we knew that through looking at the molecular details, the reason why these mutants were fat, it's because once they store fat, they're completely unable to use it. So if you put those flies through a diet, they die really fast. So they're actually highly dependent on what they eat to survive. So what we did modified diet after looking at their metabolomics and transcriptomics of enzymes that gave them the nutrients they were able to use, and that rescue a lot of phenotypes. So again, I'm not really properly answer your question. I think that basically we will have to dig in, understand exactly what are the defects that we're looking at. If indeed we pinpoint some of the defects as being a specific neuronal pathway that maybe there are known therapeutics to use, if these neuronal pathways overlapping with something else like sleep, for instance, maybe we can use some of those common therapies. But I think it's still really early in order to really extrapolate and try to come up with solutions on how to do these. And that's exactly what excites me about this work right now.
That's great. We've had 2 questions come in through the chat. If you can answer them within a minute, that would be great. Otherwise, then we're going to have Joe go ahead and introduce the next speaker. So I'll give you about a minute to answer these or address them. Otherwise, maybe you can email the question.
So I'll go quickly through Isabel's question. Is that comment on the role of ARC on RNA transfer, viral capsid-like phenotype? If this could be a mode of regulation. So we're looking into these because I showed you the amounts in the brain. They go high in low calories. They go down in the storage tissue. So what we see is that when the brain is raising levels, the storage tissue is doing the opposite. One interesting thing that we see that it's very uncharacteristic for the fat cells is that this upregulation of ARC in the fat cells is very patchy. So what we're looking into is potential projections from the brain to the fat cells that could be delivering these capsids. So this is an exciting mechanism that it's been only shown. It was shown both for flies and mammals, but in between neurons and neuromuscular junction, not yet with fat cells. So we're trying to look if that could be a mechanism. The other one is mutations are present during development and not only postnatal. They're present. The mutations are present all the time. Through some of the diet rescues that we've been doing, we can have different effects. So we haven't worked much in turning on and off mutations yet, but we have the ability to test that. So to try to test if this is prenatal or postnatal, it's required for development or only in adulthood. We haven't played much with the different stages of development, but that's something that I think it will be probably very important for some of them, not necessarily all. One of the mutants that we worked on, it had the same effect in all the different stages in development independently of where it was turning on and off. Okay, thank you so much for the opportunity. I'll pass it to the next speaker.
Great, thanks so much. This is Joe Frank. I am happy to introduce our next speaker this morning, Dr. Kathleen Gavin. Dr. Gavin received her PhD from East Carolina University in 2012 and completed her postdoctoral fellowship in the Division of Geriatric Medicine at the University of Colorado School of Medicine. In 2018, Dr. Gavin was named a Betro Foundation Early Career Investigator, and in 2019, she was selected as one of the Department of Medicine's Rising Stars. Dr. Gavin studies the role of adipose tissue and adipocyte lineage and chronic disease risk in aging women and men. Her talk today is titled White Adipose Tissue, Not So Simple After All. Dr. Gavin, thanks so much for sharing your work today.
Thank you so much for the invitation. Let me just get my slides up here. Okay, so thanks everybody and good morning. Let me get this sharing. Okay, so I'm going to talk to you today about white adipose tissue. So I think adipose tissue is one of the most interesting organs, and one of the reasons is because it has an extraordinary capacity to shrink or expand. So we're all familiar with the epidemic of obesity and how it's associated with a number of chronic diseases. But what many people don't appreciate is the other end of the spectrum of lipodystrophy. So this is a syndrome where people are unable to actually store fat in the depots that they usually store it in. And this is also associated with a number of chronic disease type problems. So hypertriglyceridemia, insulin resistance, hepatic steatosis, among a number of other things. So really both extremes have serious health consequences. And this really just demonstrates the importance of adipose tissue in maintaining physiological homeostasis and the ability for us to store the proper amount in the proper places to help maintain health. So it's important to note that we now recognize adipose tissue is an endocrine organ. So it isn't just an energy storage sink. It actually produces a number of different secretions that you can see here on the left side of the screen. And all of these factors act both locally in the adipose tissue as well as throughout the body to regulate things such as insulin sensitivity and glucose homeostasis, as well as appetite and energy balance, angiogenesis, and inflammation and immunity, among many other things. So what I'm really interested in my research is how the location of adiposity is really important in disease risk. So we know storing excess adiposity is unhealthy, but what we also recognize is the fact that people come in all different shapes and sizes. And so I'm really interested in women's health. And so women in particular really have specific body fat distributions. When they're young, in the premenopausal age group, they tend to store their fat in the lower body regions and more subcutaneous depots just under the skin. And this body fat distribution is really protective against chronic disease risk. So young women have a lower risk of cardiovascular disease and diabetes. As women age, they tend to shift more towards an abdominal fat distribution. And this is characteristics of males as well throughout their life, where they store more fat in the abdominal region and in the visceral compartment, deep in between the organs. And we know that this body fat distribution is associated with increased disease risk. And so there's a number of different hypotheses why storing fat in these different depots may be related to chronic disease risk. But what I really focus on is how the cells that comprise these adipose tissue depots may actually differ and thereby lead to differences in their overall metabolic phenotype. So I want everyone to understand the fact that adipose tissue is not just a large depot full of fat cells, but it actually has a heterogeneous composition made up of a number of different fat cells. And although by volume, adipocytes or fat cells make up the primary composition of fat tissue, the stromal vascular fraction of non-adipocyte cell types makes up the majority of the actual cell numbers within the fat tissue. So the stromal vascular fraction is made up of a number of different immune cells, as well as mesenchymal stem cells, blood vessels, endothelial cells, and neural innervation. So all of these things combine to make a really wide-ranging composition of adipose tissue. What I'm really interested in understanding is how there also may be differences in actually the types of fat cells that are in the major white fat depots. And I just want to pause and say there's also brown fat depots in humans. I'm not going to talk about those at all today, but we're just going to focus on these major white fat depots that you generally find in the subcutaneous and visceral regions. And I'm interested in understanding if there are different types of mature fat cells in these white fat depots, and if so, where do they come from? So we know that fat cells turn over in humans at about 10% per year. So even if you're not gaining or losing weight, your body is always needing to produce new fat cells. So there's a sink of mesenchymal stem cells that are actually located in your adipose tissue that are there and ready to take up the signal when your body needs a new fat cell. These mesenchymal lineage stem cells commit to becoming an adipocyte precursor or pre-adipocyte and then differentiating to a mature adipocyte. So conventional dogma has long held that all new fat cells arise from this mesenchymal lineage adipose tissue resident development pathway. Well, just over 10 years ago now, an investigator on our campus in the Division of Pulmonary and Critical Care Medicine, Dr. Dwight Clem, discovered that in fact, the story might not be so simple. Mesenchymal lineage cells might not be the only lineage contributing to adipogenesis or the generation of new fat cells. And so what he discovered is that there's actually an alternative adipocyte development pathway. So he is a basic scientist and did all these original studies in mice. But the overarching theme is that he found that hematopoietic lineage cells arising initially in the bone marrow, so these are CD45 positive cells that myeloid lineage cells actually circulate through the blood, end up in the adipose tissue. And we know that this is true because we've known for years that the stromal vascular fraction contains myeloid lineage immune cells. But what he found that was really exciting was that these cells could actually undergo an unexpected mesenchymal transition, committing to the adipocyte lineage. So becoming what we've termed a bone marrow derived precursor, ultimately differentiating into a bone marrow-derived adipocyte. And again, to clarify, these are cells that start their life in the bone marrow, traffic through the blood to the adipose tissue and are actually becoming adipocytes in the white adipose tissue. So this was really interesting. And so when I came in as a postdoc fellow, my first interest was, okay, these cells, well, first, sorry, I'm skipping ahead. Let me tell you a little bit about these cells and why we think they're so interesting and important. So let me review here that bone marrow derived adipocytes start their life in the bone marrow. They travel through the circulation to the adipose tissue where they can undergo mesenchymal transition, differentiate into adipocytes, and then accumulate over time. So the longer someone lives, we think the more that they produce of these cells. So what are the characteristics of these cells and why do we think that they're important? Well, Dwight has done a number of experiments in his mouse models to understand the characteristics of these cells. And one of the most interesting things he's done is a gene array. So what he did was he was able to separate out bone marrow derived adipocytes from conventional white adipocytes. And he found that using gene array, there's a number of different inflammatory cytokines that are highly divergently expressed between BMDA and conventional cells. So the inflammatory cytokines are highly increased expression in the BMDAs compared to conventional. He also found that leptin, the classic factor that's secreted from adipocytes, was actually very low in BMDAs. And this is interesting because leptin is really an appetite regulating and has some role in energy metabolism. So really interesting there. Some data I'm not going to show. Also, he found decreased expression of mitochondrial enzymes in BMDAs compared to conventional mesenchymal white adipocytes, demonstrating that these cells might also have some differences in their energy capacity. So really showing that maybe these cells have a harmful inflammatory metabolic phenotype. One other thing that really interested me was the fact that BMDAs were present in all of the major white fat depots that he's measured in the mice, not in the muscle, liver, or lung, as we would expect, but really their highest production are in some of these depots where it's more harmful to carry your fat. So the fat depots next to the heart and the gonadal fat depot, which is a visceral depot in the mouse. So very interesting to see that these fat cells may potentially have a harmful metabolic phenotype and are being produced in depots that are also associated with increased disease risk, at least in humans. So I was really interested coming in to really determine the clinical relevance of BMDA. So I have a clinical research background, and when I came in and started working with Dr. Clem, I was able to kind of take the reins and run with trying to understand these cells in humans. So what we were able to do is collaborate with the bone marrow transplant clinic here at the University of Colorado Hospital, run by Jonathan Gutman. And one of the major challenges that we have in studying BMDAs in humans is the fact that the cells, once they become mature adipocytes, actually lose the markers that they had saying they came from the bone marrow. So the CD45 and CD14 positive markers that usually identify these hematopoietic lineage cells actually are no longer present once they are committed to the adipocyte lineage and become a mature adipocyte. So unlike in animals where you're able to use a number of lineage tracing strategies, bone marrow transplantation with fluorescent or luciferase tags, in humans, you can't do those things. So we were able to collaborate with the bone marrow transplant clinic and recruit in patients who had actually had bone marrow transplants for hematologic malignancies. So these were patients who were doing well, at least 6 months to about 4.5 years past their bone marrow transplant. And what we did was actually use a chimerism analysis. So we brought these patients in, took fat biopsies from their abdominal subcutaneous adipose tissue, isolated out the fat cells, and then extracted the DNA. Then we were able to use the same clinical measurement of chimerism that's used for engraftment assessment purposes in these patients and find that there was actually evidence of some donor DNA in the fat cells from these hematopoietic stem cell transplant recipients. So you can see here on the graph, we have months post-transplant on the x-axis and the percent donor chimerism on the y. So anywhere from 5 to 35% of the fat cells that we isolated actually demonstrated evidence of donor DNA, demonstrating that some of these cells arose from the stem cells that were put in through their hematopoietic stem cell transplantation. And the colored dots here just indicate patients who we brought in for repeated fat biopsies, anywhere from 6 months to a year after their first biopsy. And in all of them, we saw an increase over time in the production of their BNDAs. So this was really exciting. And we didn't know, but at the same time we were doing this, a group, a really well-respected group in Sweden was performing the exact same study in a larger cohort of bone marrow transplants participants, and they found the exact same results, that less than 5 to 27% of the subcutaneous adipocytes they isolated seemed to be coming from bone marrow derived sources. So this was really exciting, corroborated by another lab who was working completely independently, and really helped to resolve a little bit of the controversy in this field. Traditionalists in the adipocyte biology field did not want to believe that hematopoietic stem cells could actually be part of this adipogenesis. And so having another group independent to us publish, especially in a high impact journal such as Cell Metabolism, was really exciting. So one of the challenges that we have in continuing to study these cells in humans is the fact that these hematopoietic markers disappear on the mature cells. So unless we're using a population like a bone marrow transplant patient, we aren't still able to measure these in humans of anyone who's walking down the street. So I've really been working on searching out a biomarker to try to determine how we can measure these cells in humans. But in the meantime, I've actually done most of my work in culture in developing a human primary BMDA cell culture model. So what we do here is we're able to obtain fat biopsies from healthy research volunteers. We obtain their biopsy, we collagenase digest the sample, we spin it out to pellet the stromal vascular fraction, and then we can stain them for flow cytometry. And so we use FACS in order to identify the different cell populations that we get in that stromal vascular fraction. So we're able to identify our conventional mesenchymal lineage adipocyte precursors, and this is just an example of our flow cytometry gating strategy. But here's our 45 negative population, which we then will take and have a few more steps of identifying other surface markers such as CD29 and CD34 to identify this population. But we can also identify adipose tissue macrophages, monocytes, neutrophils, and lymphocytes, and really clearly and robustly, repeatedly sort these out and be able to put them into a cell culture model. So what we do is we take our mesenchymal lineage cells, we sort them out, and we plate them on plastic according to traditional mesenchymal stem cell culture. So we proliferate them and expose them to different adipogenic media, and they differentiate into fat cells. Now, for those of you that are familiar with hematopoietic cell culture, they tend to not grow adherent plastic. So they grow in suspension culture. And so what we were able to do in combination with Dr. Clem's lab is actually generate a 3D cell culture model in which we resuspend the hematopoietic lineage cells in a 3D fibrin matrix. So we suspend them in this matrix for 5 to 7 days, after which we digest the fibrin, recover the cells, and during that time, we found that the cells in that matrix are undergoing a hematopoietic transition, whereby which when we remove the cells from that fibrin, they actually become plastic adherent. And they will proliferate and grow similar to conventional mesenchymal lineage cells. So we've used this model, and the last couple years we've been working on really characterizing it and working on getting this model published once and for all for the human primary BMDA cell culture. So I'm going to show you a little bit of that data now. So we found that the BMDAs, when compared to conventional adipocytes, proliferate at an identical rate as measured in the live cell imaging IncuCyte. So that's really neat. So when we plate them on plastic at the same time, they both grow similarly. We've also found that whether we're looking at conventional lineage or a myeloid lineage cell such as a neutrophil that we've isolated out, they both proliferate and differentiate to a similar capacity. This is a bright field image on the left. This is an oil lipid stained image on the right, and you can see that both differentiate. I also want to note that we've actually isolated out some hematopoietic from a blood draw. We've stained them the identical way as our stromal vascular fraction from fat, sorted them through FACS, and put them into our 3D fibrin culture, and actually find these cells do not grow at all. They don't proliferate or differentiate. So there is some signal in the adipose tissue telling these bone marrow derived cells to go down what would normally be an unexpected pathway. So we're really still working on trying to figure out what exactly are those signals in the adipose tissue telling these cells it's okay to go down the mesenchymal lineage. We've quantified the differentiation of these different lineages with oil red O. We find the differentiation capacity between conventional or macrophage monocytes or neutrophils are all similar. And there's a number of bar graphs here, and the units don't really matter, but what I want to demonstrate is no matter what major adipocyte protein that we've looked at in these adipocytes, we find that after 14 days of differentiation, you see a significant increase in the major adipocyte proteins and to a similar level in conventional and bone marrow derived adipocytes. So whether we're looking at PPAR gamma, CEBP alpha, fatty acid binding protein, CD36, we see in all of these cases, these adipocyte proteins are present in BMDAs as well as conventional adipocytes. So these cells do seem to be differentiating into bona fide adipocytes. We're also trying to interrogate the metabolic phenotype of these cells. And we're just at the beginning of these studies, but one bit of preliminary data that I was able to share with you today is the fact that we've looked at some of the factors released into the conditioned media. And we have indeed found that there are a few factors that already, even with a small N, in matched conventional and bone marrow derived adipocytes from the same person, we're seeing that IL-8 is secreted to a greater extent in the bone marrow derived adipocytes as compared to conventional mesenchymal lineage adipocytes. And interesting that leptin is actually being secreted at a lower amount in BMDAs compared to conventional. So this is similar to what Dwight found in his gene array, although our protein data show leptin is similar between the lineages. So a lot of work remains here, but we're starting to just see the signals that maybe we are able to see the same metabolic or a similar metabolic phenotype in our human primary cell culture model as Dwight is observing in his in vivo mouse models. So we have a number of ongoing in vitro studies in this area. We're really looking to determine the metabolic phenotype of human primary BMDAs. We're looking at insulin sensitivity, mitochondrial content and respiration, metabolomics and RNA sequencing to really identify maybe a gene expression fingerprint or a metabolic pathway that may be different between these. I'm also really seeking to determine a biomarker to identify BMDAs in humans. We're just embarking on some single nuclear RNA sequencing that I'm excited to get back into the lab and give a try. One thing I just want to really quickly touch on is that I have a number of different factors in mind that I'm interested in studying that may influence BMDA production, but one of the major ones that's a focus of my K award right now is estradiol. I just want to present some basic data from Dwight's mouse models demonstrating that when we look at the production of BMDAs using a luciferase tag, you can see here that in comparison to wild type, an overactimized mouse or an ER alpha knockout mouse actually have a higher production of BMDAs as compared to wild type. And adding back estradiol in the overactimized mouse actually brings the BMDA production down to lower than what it was at wild type. And this is really pronounced in the gonadal or that visceral depot in the mouse, but we're seeing similar tendencies in the inguinal fat with very low amounts of these adipocytes in the intrascapular or brown fat. So my overarching working hypothesis in the clinical realm is that the loss of estradiol, ovarian estradiol in women is related to increased or leads to increased BMDA production, specifically in the visceral region. So that's that region where we see postmenopausal women increasing their body fat with menopause, and that this may really be associated with the increased cardiometabolic disease risk observed in this population. So in conclusion, adipose tissue location and composition are primary factors in determining chronic disease risk. The proportion of BMDAs versus conventional adipocytes may be an important factor, and we're still learning more about that, especially in the clinical realm. And I really am interested in understanding if increased BMDA production may be a mechanism underlying increased visceral adiposity and chronic disease risk in postmenopausal women. So I just want to give a big thank you to everyone in the Division of Geriatric Medicine, and particularly my mentor, my clinical mentor, Dr. Wendy Court. She's been super supportive of me pursuing this research area, even though it's been quite controversial. And so really it's taken a lot of effort to continue to pursue and work against the normal flow of things, and Dwight for opening up his lab and teaching me so much basic science. And then of course, my staff who does all the cell culture work, Bogie and Ruby, are fantastic. and Tim, who works in Dwight's lab, and he has done most of the animal work that was presented in those graphs. And then, of course, the CU Cancer flow cytometry resource has been greatly instrumental in me actually being able to produce this cell culture model and identify the cells that I was looking to identify and sort them out on a really reproducible manner. And then on an unrelated note, I just wanted to give a huge shout out and thank you to everyone at the institution working so hard on understanding the COVID-19 disease, as well as all the healthcare providers on the front lines. It's difficult to continue to work on this type of research and prepare a presentation like this in the face of everything that's going on. So I really appreciate everyone at the institution, all the hard work that they're putting in and all the research that's being done to really try to help us move forward in understanding this disease. And so with that, I'll go ahead and take any questions. Let me see if I can get the chat up here.
Thanks, Dr. Gavin. That was great. Maybe we'll take 3 or 4 minutes for questions. It looks like there are a couple there in the chat box for you.
Okay. So the first question is, in the BMT patients, bone marrow transplant, do you see male versus female difference? So this is a really good question. So our sample was too small to actually do male-female comparison. I think we actually only had 2 women. The Karolinska group had a larger sample. They did not see a sex difference, but Dwight's animal data suggests that actually the female mice produce more BMDAs in comparison to the males. So I really think we need to take a closer look at that and really be able to look at pre versus postmenopausal women, and some of it in the transplant patients might have to do with age at transplant and things like that. So we have a lot to learn, but I'm very interested in the sex difference and actually have some studies going on with testosterone at the moment as well. Next question is, what about the role of testosterone to regulate the BMDA population? So I'm really interested in that, and I am obtaining samples from males. I'm really interested in the role of testosterone, or potentially the role of the aromatization of testosterone to estrogen, and the role of estrogen even in men in regulating fat mass. And so I am currently working on that. The jury's out right now. We haven't done any animal studies, although I think Dwight is getting closer to doing some orchiectomy to understand what's going on in males. I think that's all the questions that are in there. So thank you very much.
Thank you.
Okay. Thanks, Kathleen. That was great. So as Dan Polea is our next speaker, and as he starts to get his slides up, I'd like to introduce him. He is an associate professor in the Division of Hematology, the clinical director of Leukemia Services, and the Robert H. Allen Chair in Hematology Research. Following his training at the University of Chicago Pritzker School of Medicine and Stanford University, he joined our faculty at CU in 2011. Dan was selected for the Department of Medicine Outstanding Early Career Scholar Program in 2015, and today he will share with us his work translating leukemia stem cells into clinical application. Dan?
Thank you. Let me just get my video up here. Sorry. Okay, sorry about that. Can someone just tell me can you see the presenter screen, or are you seeing the main screen here?
We can see the projected slides.
Oh, okay, all right, so we're looking good. Great. Well, thank you so much for this opportunity. I'm a very lucky recipient of the Early Career Scholars Program here at this institution, and so thankful and grateful for that award, because it really is what has allowed me to do this work, and that has been a really important part of my success here so far. You know, the other thing I just want to say is in this really difficult time, I'm really cognizant of how lucky I am on a daily basis to be able to have a balance between patient care and research responsibilities, and having a big part of my daily activities taken away from me, as all of my colleagues, I'm sure, can attest, has really made me appreciate just how important this has been for my own mental health and well-being, and I'm really looking forward to getting back to the momentum that we had here, and doing something that I care about so much. What I'm going to talk about today is acute myeloid leukemia, and some of the advances that we've been having, and many of which I believe are coming from our own center here that I'm excited to tell you about. Let's see. Okay. This still works? Everyone can see okay? So acute myeloid leukemia, it's the most common acute leukemia in the adult population, about 30,000 a year in the United States. And while those numbers may seem small, the death rate from AML approximates the death rate from many other much more common malignancies. So this is a real problem. It's a disease of older patients. Median age of diagnosis is about 68. For decades, the standard of care at diagnosis has been to offer intensive induction chemotherapy, the so-called 7+3 regimen. The issue there is that this is almost never a curative therapy, and this is poorly tolerated by older patients. And of course, older patients make up the vast majority of patients with this disease. So we have a standard of care in this disease that doesn't apply to the average patient with this disease. And as a result, over the last many decades, where there have been no advances in treatment options for these patients, the outcomes have been just abysmal. Large numbers of patients at time of diagnosis aren't even offered therapy at all. And so, clinical investigators have been wondering for a very long time, are there other ways to do this? Do we have other options? And one potential opportunity is to think about targeting the leukemia stem cell population. Leukemia stem cells are the small subpopulation of this disease that we define as having self-renewal and proliferative capacity. And I've been here now almost 9 years, and I was very fortunate after the first several years of being here to be joined by our division chief, Dr. Craig Jordan. Dr. Jordan is a worldwide expert in the biology of leukemia stem cells, and I was very familiar with his work even as a fellow. In fact, the first Journal Club paper I ever presented was Craig's paper on targeting leukemia stem cells using these compounds called parthenolides. So I was really, really excited about leukemia stem cells in general. And when he came to join us, we sat down to talk about how we could completely align the clinical program with everything he was doing in the lab. And if we could really be the first center to focus on clinically therapeutic targeting of leukemia stem cells. And when we really thought about this and wrote up a couple of review type papers about it, we thought there's huge opportunities here. If you can eradicate the leukemia stem cells, you can eradicate the population of cells that are resistant to traditional chemotherapy. And this can give you the potential for deep and durable remissions, irrespective of cytogenetic or molecular risk factors. All those lesions would be expected to come after the leukemia stem cell, the genesis of leukemia stem cell. Challenges here, though, were great because there was not a whole lot of consensus on how to even recognize, define, or identify leukemia stem cells. And also, leukemia stem cells have a great many similarities to normal hematopoietic stem cells. And if you exploit a weakness in a leukemia stem cell that's shared by a normal hematopoietic stem cell, then you run the risk of ablating the bone marrow. And as I like to say, my colleagues like Dr. Gutman are very good at ablating the bone marrow in the setting of bone marrow transplants. I would prefer not to do that. And so you have to find this sort of silver bullet that can exploit a weakness in a leukemia stem cell that's not shared by a normal hematopoietic stem cell. So over 10 years ago now, it was first, the concept of targeting BCL2 as a potential way to target leukemia stem cell was first introduced. BCL2, of course, is a member of the BCL2 family of proteins, this large family of proteins that's responsible for the equilibrium between apoptosis and protection from cell death. BCL2 is the protein that's the main protein responsible for anti-apoptotic or protection from apoptosis. The Jordan lab published this seminal paper in 2013, first showing that you could identify leukemia stem cells by the environment in which they reside. In this case, the Ross-Low environment characterizes where leukemia stem cells live. And when you do gene expression profiling, you can see that one of the most upregulated genes in the leukemia stem cell compartment was, in fact, BCL2. And taking that to its logical conclusion, if you used a BCL2 inhibitor, a much less selective one than we have available now, you could eradicate the leukemia stem cell compartment. So in the next 10 or so minutes of the talk, I want to go through this mechanism for how a BCL2 inhibitor can target leukemia stem cells. So let's just sort of build that, starting with this slide here. This is the first evidence that, yes, a BCL2 inhibitor could target leukemia stem cell population. Back to this seminal paper, I just want to quote it here. I think this is pretty useful because this was a pretty big deal. In contrast to normal progenitor cells, LSC-enriched subsets may paradoxically depend more on mitochondrial respiration rather than glycolysis to meet their energy demands and maintain their survival. So this was a big deal because this went against a phenomenon that had been a sort of observation for many decades in cancer biology, the so-called Warburg phenomenon, that cancer cells preferentially use glycolysis over oxidative phosphorylation for their as their preferred metabolic energy source. Here we saw that in the setting of leukemia stem cells, and has now been recognized in many other cancer stem cells, the citations for those are listed below, it's the opposite. These are dependent on oxidative phosphorylation, and they prefer that source of metabolism over glycolysis, which was a big difference. The Jordan lab went on to show that when you use a less specific BCL2 inhibitor than the one we have now available, you can see that in different cell types, in this case, total AML, the Ross-high population, application of the BCL2 inhibitor could decrease oxygen consumption, the readout for oxidative phosphorylation. That was also true with respect to the Ross-Low leukemia stem cell population and even in the normal hematopoietic stem cell compartment. The big difference was when you look at compensatory glycolysis, all these other cell types could compensate just by upregulating glycolysis, whereas the leukemia stem cell population was unable to do this. That was a unique property that differentiated this leukemia stem cell population from all these other populations. It perhaps could be thought of as a way to target this population. And then when you look at the readout for decreased metabolism, ATP production, you could see that applied in the Ross-Low leukemia stem cell population, not so much in the bulk disease, and not so much in normal hematopoietic stem cells, unless you also did something to impair other forms of metabolism shown here. So when we had an opportunity a few years later here at our institution to participate in a clinical trial of newly diagnosed, unfit for induction chemotherapy, older AML patients using venetoclax, a very specific BCL2 inhibitor, we enthusiastically participated. The study design used a hypomethylating agent called azacitidine in addition to the venetoclax. And the initial experience here at University of Colorado was we enrolled 33 of the patients. Really, what we're showing here is just that these patients had adverse risk features. This was not a good risk population. But the upshot of this experience was that patients treated with the venetoclax plus azacitidine regimen compared to patients treated in our practice over a 10-year period with in a similar age population with really any other treatment, be it intensive induction chemotherapy, low intensity therapies, clinical trials, there's a very significant improvement with respect to overall survival. So it's very exciting, and we were sort of cued into this pretty early on through participation in the clinical trial. Over the years, we've had the opportunity, and it hasn't been that long, but over the last several years, we've had the opportunity to treat nearly 200 patients at this institution with this regimen, and we've learned a lot about it. This is one of our recent clinical papers looking at this treatment regimen in 3 different settings. So the first is what we call the ideal world. This was the clinical trial with all the usual clinical trial parameters and restrictions, and you can see that in that setting, overall response rate of 91%, complete remission rate of 85%. You know, by contrast, just using azacitidine alone, we would expect only about a 30% response rate, and that was the sort of de facto standard of care for many years for patients who weren't good candidates for intensive induction chemotherapy. But there was this strange phenomenon going on because venetoclax had been a few years before this approved for a different type of leukemia, CLL, and so we had access to venetoclax off-label at the same time that we had the clinical trial up and running. So patients who didn't meet the eligibility criteria for the clinical trial, we could go ahead and put onto the exact same regimen with off-label use for most of the patients. And this is what we call the real-world scenario. And you can see in the real-world scenario, you know, response rates were lower. Now, these patients, some of them on dialysis, some of them with other active malignancies, so real-world scenario, but still much better outcomes than we would have expected with just azacitidine alone. The third scenario we looked at was what we call the fantasy world. A lot of people thought that the real comparison should not be, for this regimen, should be patients who could get intensive induction chemotherapy. We, of course, felt that that was too toxic for most of the patients, but we were able to use this online verified database where you could plug in baseline characteristics of patients, and this would spit out the expected response rate and toxicity or death rate from induction chemotherapy. And when we did a statistical comparison between our patients who got the venetoclax regimen and what would have happened in this fantasy world where they were treated with intensive induction chemotherapy, we found about the same response rates, but a significantly decreased rate of early death or therapy-related toxicity with the regimen, with the venetoclax regimen. So all that just led to our suspicion, our strong suspicion that this was a superior regimen. So back to mechanism, we were very keen to sort of figure out whether we could show using in vivo patient samples similar mechanism that Dr. Jordan's group showed with in vitro work from several years before. And so here's where things got difficult, because as a clinician, I was very excited to see, you know, a 90% response rate, and the responses happen very, very quickly. Within 28 days, most patients are in a remission. And when I took that to my laboratory-based colleagues and said, hey, let's try to figure out using these post-treatment samples how this is working, they looked at me and they said, well, there's no disease cells left. There's no leukemia cells left. We're not going to be able to really tell you very much. And so we sort of rejiggered things to start to think about how can we look at patients' responses almost in real time within a couple days of treatment. And this is where studying leukemia is so nice, because many times leukemia circulates in the peripheral blood. And so we really started to focus on those patients who had circulating disease. And you can see here that doesn't last long. This regimen works so quickly, most of the circulating disease is gone within a few days. But we thought if we could really focus on post-treatment responses within just the first few days and sample peripheral blood to get the leukemia population, we might be able to really make a difference here. And so Brett Stevens, one of the research faculty in the lab, looked at some CyTOF data. You can see using a lot of different stem cell markers within just 24 hours, in fact, we were able to document that we were decreasing the leukemia stem cell population. So back to this mechanism slide, we now can show that it's azacitidine plus venetoclax that's targeting leukemia stem cells. And so we wanted to start to fill in these blanks. So going back to the in vitro work, we looked at whether oxidative phosphorylation was impacted. And in fact, in both the bulk and the leukemia stem cell compartment, we saw that very quickly after treatment, within 24 hours, there were significant decreases in oxidative phosphorylation. We also showed that in vitro, and then again, the readout for decreased oxidative phosphorylation should be ATP production, which decreased very quickly within, with treatment with the venetoclax regimen. By the way, we did not see this decrease in oxidative phosphorylation for the most part in patients treated with intensive induction chemotherapy. So this phenomenon of targeting oxidative phosphorylation seemed to be unique to this therapeutic regimen. So now we know that we're targeting leukemia stem cells and that we're decreasing oxidative phosphorylation as we would have predicted from that in vitro paper. But the next piece of the puzzle sort of came from the work of Courtney Jones, who was until very recently a postdoc in Craig's lab and has now gone to start her own lab at the University of Toronto. What she found first was that leukemia stem cells have more amino acids and are more dependent upon amino acids than the bulk disease, and that the venetoclax azacitidine regimen was impairing the uptake of the amino acids into the cell. And so because of leukemia stem cell dependence on amino acids, this was preferentially targeting this leukemia stem cell population. And by the way, you needed both the venetoclax and the azacitidine to have this impact. So which amino acids were important? She also published this. So when she cultured the leukemia stem cells without each relevant amino acid, what you can see is that the bulk disease, the cells do take a hit, but can partially compensate, as opposed to the leukemia stem cell population, which is much more sensitive to decreases in these amino acids. In particular, cysteine is the amino acid that these cells, that these leukemia stem cells really depend on, compared to things like arginine and glutamine. And so cysteine, when we sort of thought about this, it's part of a tripeptide complex that makes up the antioxidant glutathione, and this is the cysteine is the rate limiting step for glutathione production. So sure enough, when we looked at glutathione levels as expected with in vivo leukemia stem cell compartments, in the post-treatment setting, you saw nice dramatic decreases in glutathione levels. Now, glutathione regulates succinate dehydrogenase A, which is one of the critical enzymes in the TCA cycle. And it does this by this post-translational modification called glutathionation. And so when we looked at, to confirm that, yes, in the post-treatment samples, we had decreased glutathione, we also saw there was less activated succinate dehydrogenase. And since succinate dehydrogenase is the only sort of enzyme that links the TCA cycle to complex 2 or to the electron transport chain, it's through complex 2. We wanted to see if that was in fact the bridge. And so Courtney laboriously looked at all these different TCA cycle metabolites in the leukemia stem cell compartment pre and post treatment. And the X here is where succinate dehydrogenase converts succinate to fumarate. And you can see that the increase in succinate and decrease in fumarate is sort of the metabolic phenotype of deactivation of succinate dehydrogenase A as predicted. And then, in fact, she directly measured complex 2 activity and showed that with treatment with the venetoclax regimen, you decreased the complex 2. And interestingly, you could partially rescue this phenotype using cell-permeable glutathione. And so we have what is our working understanding of the mechanism here. This regimen is decreasing amino acid uptake. That's decreasing cysteine and therefore glutathione levels. You impair complex 2 the electron transport chain. You decrease oxidative phosphorylation, and you can specifically inhibit the leukemia stem cell population. So, at the University of Colorado, this is, even though this has now become the standard of care and FDA-approved therapy, we've become very inured to this. I mean, this is something that we just sort of do every day and have been doing for several years. So in the last couple of years, we've been focusing, as the world sort of catches up to how great this regimen is, we've been focusing on who's refractory to this to this regimen and really trying to understand who those patients are. And the numbers are not insignificant. While, here's a couple of different studies that have been reported, the response rates are probably around 70% when you look at the amalgamation of all these different studies. That means about 30% of patients are not responding to this regimen. So the field is really trying to figure out who these patients are and how can we identify them. And my colleagues at other centers are looking at the traditional risk factors for non-response to an AML, for AML. But that's all in the context of intensive induction chemotherapy, because that's all we've really ever done for these patients. But this regimen works completely differently. We were, ready to accept that other predictive factors would be relevant that maybe we hadn't explored. And here, these figures from recent publications or presentations show that for the most part, the traditional risk factors, they don't apply. So cytogenetic abnormalities, the ones traditionally associated with poor responses in the setting of chemotherapy, same with gene mutations, aren't really all that predictive for poor responders. Again, not too surprising to us for a regimen that works in a brand new way and can target leukemia stem cell population, but it's been a little hard for this to become widely accepted. So working with another great research faculty in the Jordan lab, Shanshan Pei, Shanshan really started to explore who were these patients who were refractory to the venetoclax regimen. And he made an observation that we clinicians miss. So this is really one of the incredibly productive ways that we clinicians have been working with, our laboratory-based colleagues in the setting of the hematology division here. We would sit in tumor board every week going over who was responding and who wasn't responding, and we were missing a very valuable, important observation that Shanshan brought up to us. He said, hey, have you noticed that a lot of these refractory patients have monocytic disease features? And so this goes back decades in AML. The way we used to classify AML was based on morphology. So an experienced hematopathologist would look under a microscope, and he or she would tell us, hey, this leukemia is arrested at a very primitive stage of differentiation, or hey, this leukemia is arrested at a less primitive stage of differentiation. And there were 8 different stages of differentiation, 8 morphologic subtypes that we understood, and that was the FAB, the French American British system. This went away because it was felt not to be biologically actionable, and it was sort of replaced by cytogenetic and molecular features. But it still exists, we just don't pay much attention to it anymore. What we found was that when we looked at 100 different patient samples, all treated uniformly, newly diagnosed AML patients, the only multivariate predictor of a lack of response to venetoclax was the FAB-M5, or the monocytic variant, or the monocytic subgroup of AML. And Shanshan further went on to make this very nice figure showing that if you have M5 disease, you're very likely to be refractory. If you have more primitive disease like N0, M1, it's much less likely that you're going to be refractory. So way overrepresented to be refractory in the monocytic subset of patients. So Shanshan went on to screen some patient samples for sensitivity to venetoclax azacitidine, and he found that resistance was very highly correlated with monocytic biology. And this work is all in vitro, so you can assume that to some degree, the resistance to this regimen here is cell intrinsic. He went on then to do a principal component analysis of the monocytic leukemia stem cell compartment versus the more primitive leukemia stem cell compartment, showed very clear differences in these populations, did a GSEA analysis, and showed that the most upregulated gene set in monocytic AML leukemia stem cells was oxidative phosphorylation. Confirmed this with a seahorse experiment showing that basal respiration of oxidative phosphorylation is higher in the monocytic leukemia stem cell compartment. So monocytic leukemia stem cells have increased energy metabolism, and it appears to be from oxidative phosphorylation. So here he showed that at the stem cell level, there's a difference in BCL2 expression in leukemia stem cells that come from more primitive AML and from monocytic AML. He then looked at the Cancer Genome Atlas data set, a couple hundred AML patients, and confirmed that in that independent data set, when you categorize by morphologic subtype, you can see that AML M5 has a decreased BCL2 expression compared to the more primitive subsets. And he also showed this at the protein level as well. And so this kind of brought us to a conundrum, because if BCL2 mediates OXPHOS and monocytic AML has less BCL2 but more activation of OXPHOS, then how can that be explained? And so we looked at other BCL2 family members. So those of you who've studied this know that there's a ton of redundancy in the BCL2 family. There's a lot of proteins that seem to have the same job. And there's the canonical function of BCL2 family members with respect to apoptosis and anti-apoptosis and pro-apoptosis, but could it be that perhaps there's some overlap here and that some of these are doing other things? So besides BCL2, the main sort of anti-apoptotic protein is MCL1. We looked at MCL1 in the TCGA database, and sure enough found that M5 has increased MCL1 expression. When Shanshan then looked at viability treating leukemia stem cells from monocytic AML populations, you can see that the blue with venetoclax azacitidine doesn't have too much of an impact on viability. But when you bring in an MCL1 inhibitor, in this case VU013 plus azacitidine, dramatic decreases in the leukemia stem cell compartment. With respect to viability, I'm not showing you here, but it applies to OXPHOS, and then the readout of all this ATP production. And this sort of important experiment to look at engraftment into a mouse model, he showed that when you look at primitive AML leukemia stem cells, the use of venetoclax-based regimens can impair engraftment into a mouse model. An MCL1 inhibitor doesn't do a whole lot, but it's flipped around when you look at a monocytic AML samples. Venetoclax doesn't really impair engraftment, but an MCL1 inhibitor does a lot more significantly. So in conclusion, what we've shown is that venetoclax can effectively target the leukemia stem cell compartment. And this is a brand new mechanism, a way to kill a cancer cell, in this case, a leukemia stem cell. And this is what's accounting, we think, for the deep and durable responses that we're seeing across genomic and cytogenetic subgroups. It's doing this by targeting the amino acid fueled oxidative phosphorylation that these leukemia stem cells have become dependent on. It's more than a preference, it's more of it's a dependence. And that is what allows us to selectively impair leukemia stem cells and not harm things like normal hematopoietic stem cells, because they can compensate. And then finally, we've shown here really for the first time that the subgroup of AML that doesn't respond to this is overrepresented by those that have monocytic disease features, that those are the patients that respond poorly to the BCL2 inhibitor. And we've shown that is at least partially because this population has decreased BCL2 expression. They're more dependent on MCL1, and they're upregulating their metabolism through oxidative phosphorylation by use of MCL1, and that MCL1 inhibition can selectively target this population. So so many people to thank, and on the lab side, the Hematology Clinical Trials Unit, which has supported all the clinical trials at this institution related to venetoclax, and so appreciative to all of them. And thanks so much for your attention. Really appreciate it. Happy to take questions if there's time.
Thanks, Dan. That was great. It looks like you already have one question in the chat from Megan Kellett. Do you see it? I can read it too.
Let me see here. Yes. Yeah, great question. This is a question I get all the time. So what role is azacitidine playing in targeting the amino acid uptake when combined with the BCL2 inhibitor? So I love this question for a lot of reasons, because first of all, in the in vitro work from the Jordan lab from 2013, it didn't suggest that anything besides venetoclax would be required to have this metabolic impact on the leukemia stem cell population. When we did this work in vivo, we found that, in fact, you really do need the azacitidine. That was a lucky accident because that was part of the clinical trial design. Exactly what it's doing and how it's contributing and why it's necessary, we don't yet know. We are working on that question very hard. It's a very important question. And through collaboration and other folks in the lab, we're hoping to understand that. But really at this point, we have some theories, but don't really well understand that.
All right, great. I don't see any other questions popping up. And so, Dan, if you can stop sharing your screen, I think what I can do is put this up and, oops, sorry. Screen. Joe, did you have any comments to make?
No, I think the slide says it all. So that concludes the first portion of our agenda. We'll take a break for 21 minutes and meet back here at noon Mountain Time for Dr. Silver's keynote address. Same Zoom meeting, same link. We'll look forward to having you all join us again at noon. Thanks.
Okay, by my clock, it's officially noon. So I think we'll restart with our final session for our Department of Medicine Research Day. Our keynote speaker today is Dr. Julie Silver. She is an associate professor and associate chair in the Department of Physical Medicine and Rehabilitation at Harvard Medical School. Dr. Silver's research and clinical work has focused on improving gaps in the delivery of healthcare services, particularly cancer rehabilitation. Additionally, Dr. Silver is committed to supporting the medical workforce through her nationally recognized research on inclusion, diversity, and equity. It is for these efforts that I am honored that she accepted our invitation to come and speak today and present her work on inclusion, diversity, and equity in academics. Dr. Silver?
Thank you so much. It's such a pleasure to be here, and I know that these are very trying times. As I put this talk together, I thought a lot about what you were trying to accomplish today and wove in some information that I hope will be helpful to everyone, not only on diversity, equity, inclusion, but also on leadership and how to do research and so on. So this is my disclosure slide, and I put it up to just remind myself to tell you that not only do women in medicine and others who do this work tend to not be paid for the work. It's also, there are very few grants affiliated with this work, and it costs us a lot of money to actually do this work. So in fact, we lose money every time we do research on gender equity, diversity, and inclusion. This is a really hard topic to talk about because people have a lot of set ideas about this. In fact, many people, regardless of whether they have any formal training in diversity, equity, and inclusion, think that they're experts. And many people like to weigh in as if they are experts. So one of the things that I want to just share with you is to really focus on thinking like a scientist and using the same criteria to measure someone who is an expert as you would if they were talking about stroke or hypertension or anything else. And that would be somebody who has published in this realm in multiple journals, done multiple studies, etc., and just kind of separate that from everything else that you think you know right now. So advocacy angles for diversity, equity, and inclusion, what I'll shorten to DEI, have taken three main angles. One is the clinical, and that's we should treat women or underrepresented minorities fairly because they will deliver better patient care. And by better patient care, that would of course mean better than men who, especially white men who are in the majority. And that really hasn't panned out to the extent that maybe people would like it to, or people that advocate for that. And I'm not actually a huge fan of that angle. I do think that people, based on their experiences, can have unique insights. But I did a whole review with my colleagues on this. And you can see from the review, if you read it, that that angle, that certain groups are better than other groups, probably isn't the strongest angle. So another way to approach this is an ethical angle. And just say, listen, we need to treat people fairly because it's the right thing to do. And that's a pretty reasonable angle to take. The third one is financial. And this is by far your strongest angle. So if you're going to advocate, if you can make a financial argument then you're going to have a much stronger, or a much easier way to get things done. So you're going to see a lot of financial arguments in my presentation. So let's put our scientific hats on and make sure that we think like physicians and scientists, and just take a look at a problem that is ubiquitous and worldwide. So this problem is called queuing. And this is where women stand in line to go to the bathroom. Now, it's really an interesting problem because we can see very clearly here that from the data that there are differences between men and women. And the result is that women stand in line for long periods of time. Now, women have time poverty, and that's been shown in lots of different studies, that women spend more time taking care of kids and doing household chores and things like that, and they have time poverty. And so it really doesn't make a lot of sense for them to be spending time standing in line. There's two approaches to this. The first approach is to fix the women. And that's an approach that a lot of people advocate. And that starts with women should. So women should stop menstruating. Women, we can do that hormonally, surgically, etc. Women should not take their children to go to the bathroom. That would be another women should. So there's a lot of ways that we could say women should change their behaviors, women should change their physiology in order to accommodate the structure, the physical structure, and move the line faster. Now, another way to approach this problem would be to change the structure, and that's called structural bias, or lots of different terms that we can use, organizational bias and so on, that have been studied. And in that model, it would be more like leaders should. So if we said leaders should, we would say leaders should make bigger bathrooms for women. Leaders should do a very simple calculation. That's a design thinking calculation, which is just figuring out how long people take to go to the bathroom and just adjusting the number of stalls and bathroom space and so on for that. So leadership is a very important way to approach this. And so anytime I hear somebody saying women should, I instantly recognize them as people who don't really use data, and leadership as people who tend to use data and understand structural bias and things like that. So when we're talking about these issues, one of the things we want to do is avoid critical thinking errors, because that's really what drives the inequities that we see, that are slow to change. And so this just lists some of the critical thinking errors that are very common and easy to spot once you kind of get used to it. And a very common response to any problem with a diversity issue, with a DEI issue, is let's form a task force and have that task force address it. And one of the interesting things about forming task forces is that it probably has made things worse rather than better, because it has reassured leaders that something is being done when in fact things might have gotten worse. And the only way to really understand if things are getting better or worse is to use data. So the task force itself isn't a problem. The problem is when the task force is not using metrics to drive change, when the task force only develops a policy statement and has no idea whether that policy statement is being implemented, or only makes suggestions about something and has no idea whether those suggestions are even being taken and implemented and so on. So as we move into thinking about data, let's just focus for a minute on academic medicine. And actually, a lot of this talk is focused on academic medicine because you all are in academic medicine. So recognizing 4 gatekeepers in academic medicine is very helpful to understanding how anyone who comes from an underrepresented group may not be successful in academic medicine. Now, remember, I said I'm always going to make a financial argument if I can. So I know there's leaders who are listening to this, and I'm going to say that institutions spend a lot of money hiring diverse faculty. They recruit diverse faculty, they hire diverse faculty, and so they're putting lots of money into diverse faculty. And if they don't treat the diverse faculty well and fairly, that's a problem. But also, if the 3 other gatekeepers don't treat them well and fairly, that's also a problem. So the return on investment for chairs and deans who are hiring diverse faculty, for good chairs and good deans who are doing the right thing, the return on the investment is not good for them. It's not a smart investment for them if the other gatekeepers do not treat them fairly. So let's look at that. This is gatekeeper number 1. This is your home institution. How does your home institution treat you? One of the things that that we know in gender equity is compensation is top of mind. So top of mind is gender equity, and we have all these studies that show that women are not paid fairly for their work. And as I have done this research, one of the things that just surprised me, and I give talks and so on, and often at the end of talks, someone will stand up and say something, and it really goes against the research. And sometimes we call them pay gap deniers, or people who are just resistant to, understanding the research or thinking about it or whatever it is. But as I started to think about this, I started to think, well, maybe they really don't know the research. Maybe they really don't understand it. Maybe what they're saying is so odd and so not consistent with the research because they don't understand the research and they haven't really seen it. So as I started to think about that, I started to think, well, how can we show that women are primarily producing this research, disseminating this research, talking about it, and that men who are primarily the leaders, the deans and chairs, and by the way, the chairs in medicine I looked up just before I signed on, and it's about 80% of internal medicine chairs are men, so 80%. So the way I did this study to show that, and this is actually an important teaching point for anyone, is that you can have a question in your mind and almost always figure out a way to present it and to think about it. So I wanted to show what I thought was true, which is women are primarily doing this research, they're primarily disseminating it. It's sort of an echo chamber, and that's a big disconnect if men are primarily making decisions. So what I did in this study is my colleagues and I, we looked at all the compensation studies that were done recently, and we looked at who the authors were. Then we looked at who was citing this research in other reports. And then we looked at who was disseminating this research on social media, especially Twitter. And of course, what we found was what we suspected, which is that women are the primary people who produce it, disseminate it, and also that most of this research was not funded. So that's a really important point, and that brings me to this idea that you can use in anything you do, whether you're a leader or whether you're doing research on high blood pressure or diabetes or anything else, is you can think through this model of adoption that was started actually with technology, but it's really helpful to think about it for anything. And this model of adoption basically has producers, the innovators doing the work, and then disseminators disseminating the work. And then the chasm actually is where most startup companies and so on fail, or where most ideas fail, because you have this small group of people who believe it and understand it and agree with it and so on. And then you have to cross the chasm and bring it to the early majority and then the late majority, and then really never bring it to the laggards. And that's something to understand, that some people will never get on board. They're just never going to get on board. Whenever I give a talk like I'm giving today, I'm primarily talking to the people who are in the early majority and the beginning of the late majority. And I craft every talk in that way because I know that there's already going to be a bunch of people who are early adopters and believers and so on. So I don't really need to convince them. What I need to do, though, is convince people who are on the fence and need more data that this is real and this is a problem and we need to do more. If I spend a lot of time trying to convince the laggards, the people who will never be on board, that is just a waste of time. And this has to do with a lot of business research and so on that looks at change and organizational change and things like that. So you can use this model to really help you figure out who you're talking to, how to craft your message, and how much time to spend. So as you think about that, I put this up because a lot of my research is not in gender equity. I did gender equity research to help our faculty members as I saw problems with what we were doing in terms of recruiting this very diverse faculty and not having them have support. And my real research is in cancer rehabilitation and prehabilitation. And so one of the things that I just wanted to share with you today, again, is that you can use these concepts I'm talking about today on any type of research to help translate your message. This is a perspective that I wrote that just came out today in the British Medical Journal. And you can see that one of the things I'm talking about here is how prehabilitation could help save lives in a pandemic situation. And I discuss in this information about prehabilitation, what exactly it is, and how when we put people, when we have stay-at-home orders or whatever, they become cardiovascularly deconditioned, et cetera, that could be quite devastating for them should they have a coronavirus infection. So again, you can use the same kinds of concepts I'm using with DEI in your own work. So let's talk about compensation. Let's actually look at some of these studies. Now, this study was just done and published in Health Affairs, and Losasso is actually a professor in economics. And I like this study because I think it really highlights a few key points. One is that there's this large body of literature that documents compensation disparities, and that pay gaps persist even after you look at all these covariates, et cetera. So in this study, what they did is they looked at a group of graduating residents in New York City. And these residents were going to all different places in the country. And so they looked at their gender differences in starting salaries for the past 2 decades. They looked at work-life balance, and they looked and they did some very fancy statistics. So this is really an excellent study and really well done that shows these things. And when you look over to the right hand of the screen, you see in the turquoise that those are the unexplained. Those are the disparities that just can't be explained even after you look at covariates, including debt and specialty and all these other things. There's still this big chunk that you cannot explain. And that is where the idea comes from, and it's not just an idea, it's proven science, comes from we pay women unfairly right from the get-go, and that keeps going throughout their whole careers. And so this is a study looking at department chairs at public medical schools and noting that women chairs are paid 88 cents on the dollar as men. Very, very interesting. So women get paid less right from the start, and then that carries out throughout their entire career. So let's take a look at that. First of all, this is another study by Jenna et al., and they looked at US public med schools, and they found, for example, in neurology, that the pay gap after adjustment was about $30,000 a year. And I'm going to show you what that translates into over a career. But before I do that, I want you to note that in radiology, men were paid less. And that can happen as well. When you don't use data and you're not doing things fairly, then men can also experience disparities. And so that's a really important part, especially as more women ascend to leadership positions. We want to really think through what is a fair way to treat everyone, including all of the men, as we develop our own compensation strategies and so on. We really don't want to do what's been done in the past because it hasn't worked out very well, and we should aim to be ethical and fair, even if we were not treated fairly ourselves. So consequences of a pay gap, and this is a very, very conservative estimate that if, over the time, women would have millions more, very conservative estimate, and I can show you more, other estimates that are much higher, but no matter what, a small pay gap ends up costing women millions. So looking at, this is a study that looked at differences in salaries of physician researchers. And this was mid-career versus early career. And the first study had, the pay gap was 12,194. And the second study, they refined it a bit. And this is Dr. Reshma Jagsi who's doing these. And this was a really, really well done study. It's one of her favorite studies because of how well it was done and how the covariates were analyzed and so on. And the second one included spousal employment, et cetera. But again, you can see that even after accounting for covariates, women are paid less. So here's what is not best practices. Asking people to negotiate. That's really not considered best practices these days. People doing hiring need to understand that there is this corrosive impact of salary inequity on faculty morale, and that we want to avoid even small inequities, because these grow over time. So that's a really important thing to understand, and also increasingly becoming illegal to pay people differently for the same work. And that's true in Massachusetts, where I live and work, that a new law was just placed to make this illegal. So let's move away from salary for a minute and just talk about RVUs, because people will say, oh, but you know what? They're billing the same RVUs, and so we're paying them fairly. Well, what if RVUs aren't fair to begin with? What if in urology, you get paid a lot more because there's more men in urology, men physicians. You get paid a lot more to do a biopsy of a penis than you do in OBGYN to do a biopsy of a vagina, even though they're almost the same exact procedure and technical skill and so on. What if the basis for RVUs isn't even fair? And that's what this study showed. So to basically say, no, women and men get paid fairly because they bill the same number of RVUs, that's assuming that RVUs are fair to begin with, which they are not. And so now let's talk about things like, all right, let's add some extras. Let's give people a bonus if they get really good patient satisfaction scores. So when we look at patient satisfaction scores, and this is also true of peer assessments and also supervisor assessments. So there's research on all of these things, looking at patients evaluating women and ethnic and racial minorities, peers evaluating women and ethnic minorities, and supervisors evaluating women and minorities. And almost all of these studies show that women and ethnic and racial minorities in particular, because that's where most of the research has been done, but it could also be across the gender spectrum and can include some other very interesting things. But these studies show that in general, people from underrepresented groups are rated as less smart, less adequate, etc. And in this, by PGY3, this was a review of the literature. By PGY3 year, male residents rated higher than female residents in all 23 milestones. Now, how can that possibly be anything except bias? I mean, there's just no way to conclude that, men are just categorically better in all 23 milestones in 33,456 evaluations. That just doesn't make any sense at all. So an interesting study looked at, well, how do we switch this? And so in this study, what they did is they looked at teacher evaluations. And by the way, this starts way back in when you look at ratemyprofessor.com and you look at the analytics behind that. Women professors get rated worse in almost every category than men. And so it starts way back. So in college, what they did in this study is they basically put in the teacher evaluations just a statement before the students took the evaluation that gender bias and bias against racial and ethnic minorities is very common, and to please rate the professor fairly. And just with that simple statement, just a simple heads up their evaluations changed in favor of the women instructors. Really, really interesting study, and just absolutely fascinating at how simple a little bit of heads up, and that has to do probably with implicit bias, which is unconscious bias, that people aren't trying to be unfair. They're just so conditioned to believe and think that men actually are a little bit smarter, a little bit better, a little bit funnier. They just are so conditioned that that's true, that they don't even think they're being unfair. And then when you bring it up and you say, wait a second, maybe women are just as funny. Maybe they are just as smart. Maybe they are just as good of a teacher. Then all of a sudden things changed. So when you start thinking about implicit bias or unconscious bias, one thing that you can do is put a letter recommendation into the gender bias calculator. And that could be a letter that you write, or it could be a letter that was written about you. And you could see how much bias there was in that. So one of the things I just encourage people to do is be part of the solution, use their voice to amplify the work. Think about that every single time you fill out an evaluation. Be really careful. Somebody is reading that evaluation and somebody's judging that person based on what you're saying. And if you're really harsh and if you really are not being fair, that can be very, very devastating to the individual who is the recipient of that evaluation. So I try to recognize that there is bias in evaluations. I try to really respect people who have different perspectives even than I have, who come from different backgrounds than I have, and to try to really think it through before I write an evaluation. And especially for anyone who comes from an underrepresented group, I try to give them the benefit of the doubt and also say very positive things, find what is good, to sort of make up for, if you will, what I know is going to happen in many of their other evaluations. So that was gatekeeper number one. That's your home institution. Let's switch to gatekeeper number two, medical societies. So let's say, as in Mayo Clinic, you're paid fairly, and they did this study, and it showed a few little disparities here and there, but in general, they showed that people were paid fairly. But they said, look, if they don't advance fairly, even if we're paying them fairly at the same level, then it's really not fair. And Keith Lillimo, who's at Mass General Hospital, has noted, look, the number of outstanding candidates is more than adequate. We're just not filling these positions and not advancing women. So it's not a pipeline problem. It's not, oh, we don't have enough qualified women. And as you go up, instructor, assistant professor, etc., one of the things that you should know is that every school has a templated CV. And I'm sure you all recognize this, as being something that's similar to what you have. But when you have this is Harvard Medical Schools, and this is going to professor with on the clinical track. You need indentation, like I have right now speaking to you, to speak nationally. You need leadership roles, service on national committees, etc., being an editor of the journal, and all of these things, development of guidelines. And if you can't get those things on your CV, your chair can't get you promoted. So what we do is we send our faculty to medical societies, and we hope that they're able to present and sit on committees and develop guidelines to do all these things. And if they can't do that, then we cannot get them promoted because they bring their CVs back to us, and we send it to the committee. And the thing that's really interesting, again, remember, I'm making financial arguments every chance I get. The thing that's really interesting is every department will spend a lot of money to send their people over to the medical society. We spend money from our department, and we also ask our faculty to spend their money to go. And that is a really interesting thing, because when you check out how the money flows, it's the only time that money flows from our bank account and from our faculty's bank account into another place. That money flow gives us leverage. That money flow helps us to basically say, wait a second, we are actually customers of these medical societies. We can leverage that and basically say to these medical societies, our deans and our chairs are recruiting equitably, they're trying to compensate us equitably, you need to treat them fairly. There can be no exceptions. You must treat them fairly. So as I looked at this, I said, all right, I'm going to show now that medical societies are not treating people fairly, because what happened was there are almost no studies on this. So I decided, all right, I will do studies to show. Now, I used this as a tipping point. Keep in mind that I didn't plan to be a recognition award researcher. What I did was I looked at recognition awards in one society and I said, wow, a lot more men get them than women. And that wasn't equitable. And then I showed in a different society that for the past 4 years, 0 women got awards. Now, one of the things as a gender equity researcher that you realize very quickly, especially if you do research in another realm, is that reviewers tend to give you a really hard time when you submit a study. They have their own biases, and reviewers who are not gender equity experts are often invited to be reviewers, and they say all kinds of things that are just not true and not consistent with the literature. And so in this study, we had one reviewer who was furious that we showed that 0 women had not been awarded anything, and tried to call the study a qualitative study. And we said, oh no, this is a quantitative study. 0 is actually a number. That's a real number. And we used a concept that our PhD biostatistician, had known about from his biostatistics work, called the inexorable 0. And in the inexorable 0, the Supreme Court and other US courts have used that to show a prima facie inference, an on its face inference of discrimination. So if you can show a 0, the judge will on its face assume discrimination, and that flips the burden of proof from the person who's bringing the suit to the institution. It could be a store or a business or a university, to the institution to prove that it didn't discriminate. So if you basically see a 0 or near 0 number, the judge in the US court is going to say, this is an inexorable 0. It can't be explained any other way than discrimination. And unless you show me it's not discrimination, then we're going to call it discrimination. And so what I did then is I said, well, let's just go after the zeros. Instead of constantly doing, studies, I'll just go after the zeros. And this is one of the best arguments for the meritocracy argument. Whenever someone comes up and says, well, it's a meritocracy and women just don't deserve these things, and if they worked harder and so on, then they would deserve them and they would be able to advance more. When you start looking at zeros, that meritocracy argument is very, very hard to sustain. So the myth of meritocracy really dissolves under the zeros, under the inexorable zeros. So we started saying, all right, we're just going to show zeros, and showed all these different zeros. Now, what people said after we published our initial studies is they said, gee, women should nominate women. Now, again, that's the women should, right? So that, right, as soon as they say that, we know that there's a problem with their thinking, that they have critical thinking issues. So what we did is we said, okay, let's look at the nominations. And in the middle to older career, the more senior career, so mid-career and senior career awards, no women were nominated, 0 women. So yeah, I mean, people should nominate women, more women, and in particular leaders who are mostly men should nominate more women, because that's how nominations tend to come about. Deans and chairs and so on, leaders tend to nominate people in their department and so on. Interestingly though, in the early career category, women were nominated, but no awards were awarded. So that was a problem at the committee level. So even though women were nominated, they still never made it out of committee. And I know some of those folks who were nominated, and they were just absolutely stellar. And after our research came out, these women then were given awards that they deserved many years before. So one thing to just remember is for any award in medicine, there are more than 0 women who deserve it. This is just a basic concept that you can apply to almost anything. So again, we're looking at medical societies and showing that if we can prove that they are not treating our faculty fairly, then we can get them to change faster than any of the other gatekeepers because of the money flow. So we looked at presidential leadership, and we found 10 medical societies that had 0 years of women's presidential leadership. Again, looking for zeros. Then we found 4 societies that were leading the way, and that has to do with social norms research, actually, which is a whole big topic and super interesting, but I don't have time to get too much into it. But showing that it can be done and showing that there are societies that behave in a way that's different. So as we looked at this, we said, okay, we know that people are going to say, because we've heard this many, many times, women should. Women should step up to be, for election. Women should try harder and do more so that they can be elected. I mean, it was all women should. And so one of the things that we wanted to show, and we did this right after we published this sort of back to back, is we surveyed more than 1,200 women physicians, and we asked them about their prior experience with elections, and more than 40% had prior experience. So it wasn't that women didn't have any election experience. And then approximately 50% were interested in future elected positions. So there's this myth that women have no experience and they're not interested. That's not true. So when I give this talk, if I was in Colorado with you live, what I would do is I would ask the audience, how many women in this audience would be willing to take time away from their work and their families and run for an election in which the data showed that no woman had ever won, or maybe only one woman had ever won, and an inexorable zero can be a true zero or a near zero number. How many people would want to do that? And in the audience, there's usually 1 or 2 people who are very committed to breaking glass ceilings, and they'll raise their hand, but most of the other women will not raise their hands. They will just say, I'm not spending my time and energy when the data show that women are not going to win. And so I said, all right, now let's change that. Let's say that now your chair supports you, other people support you, they're going to rally for you, they're going to be part of your campaign committee, they're going to help you, and now would you run for election? And all of a sudden, all these hands go up, showing what happened in our study, which is that there's a lot of women who are interested, but they're not going to step up when they know that it's almost a surefire losing situation for them that will cost them time and money. So let's take a look at speaking, because speaking is how you build your reputation. Even giving this talk virtually, people will know in Colorado more about my work and what I've done and so on, because I had this chance to speak to you all today. And so speaking is a really big deal. And at medical conferences, it's very important. That's how you build your reputation and so on. So in the study that we looked at as speakers for medical society conferences, women were underrepresented. No surprise there, right? Women were underrepresented. By now, hopefully no one's surprised that there's, all these disparities and so on. But here's a really interesting thing. When we started to separate out women physicians from PhDs and other women, what we found is that women physicians are even more underrepresented. So even though, and we saw this in a Dean study we did, and we're seeing this in other studies. So even though women in general are underrepresented, the women that are the most underrepresented are women physicians. And that's a really interesting thing. When you kind of think things through and you say, wait a second, it goes men physicians, and then men PhDs, and then women PhDs and other women, and then women physicians at the very bottom. Why is that? Why do women physicians get the least support and the fewest opportunities? So we've been writing about this, saying, is there a reason why maybe early in women physicians' careers, maybe it's easier to mentor them, maybe it becomes more threatening or harder as women progress? What is it about that mid-career time that's making mid-career women in academic medicine, especially physicians, invisible? So again, you can be part of the solution. Anytime there's a call out for speakers or whatever, instead of saying, you know, I really don't know any women physicians who can speak on this issue. If you keep a list, and again, I don't want to only focus on women physicians because I want to support all women. Women PhDs are also underrepresented, and you know, really important to recognize nurses and just all of the women that make up the workforce. But to keep a list and to know who are the experts and always be ready to nominate someone. So micro inequities, those were the macros. Those were the big ones. The micros are small, and it's like a grain of sand. And sometimes what happens with the micros is because it's so small, the people that experience it have experienced lots of them. And so they get really angry and frustrated, and their anger often seems out of proportion to the insult. And so if you don't understand sort of the buildup of the micro inequities, then when you're as an observer, you're thinking, oh my goodness, they're just completely overreacting here. On the other hand, people who experience micro inequities should recognize, gee, I mean, am I reacting fairly in this situation to this one person and this one thing, or am I bringing everything that's ever happened to me into this situation, and I really am overreacting? So we all have to be careful about micro inequities, recognizing them and so on, but we see them everywhere. And we see them all over social media, in whether it's speaking, whether it's board examiners. On the middle picture, there's board examiners. That would be a very difficult group to face as board examiners, and so on. So NIH, the director of NIH, Francis Collins, has basically said, look, as one part of what he and the NIH want to do is they want to end MANLs. And MANLs are male-only panels. And that came out as, something in June of 2019 and sort of hit the papers, and everyone was talking about it and saying, listen, Dr. Francis Collins is not going to sit on any more MANLs, et cetera. And as the COVID-19 epidemic or pandemic has come, there's been a lot of MANLs. A lot. And it's been the subject of a lot of discussion. And this individual basically said, you know what, just because we're in the middle of a crisis, I'm not going to participate in something that I think is inconsistent with my convictions. And so he got obviously a lot of likes, a lot of retweets. He doesn't have a lot of followers actually, but this became sort of a viral tweet, because people were just like, yes, like now is not the time to start, reverting back or even increasing, disparities. Like that now is not the time to do that. So how can we put numbers around micro inequity research? You can find data anywhere and everywhere, and you can almost always figure out a way to quantify something. So one thing that I recognized, again, looking at medical societies, is one reason why women were becoming invisible is our newsletters weren't really showing their work. They weren't saying, here's Dr. So-and-so, and she does research on such and such and whatever. What they did instead was they often used women as spokespeople for the society, asking people, saying, I love this society, please join it, kind of thing. And then right next to it would be this information about men's research and work. And so what I did with colleagues, again, is we looked at how to document this, and we looked at all the articles with the photos and the names and all that in our newsletters for 5 years, more than 5 years. And we found that more than a third of the time, women weren't mentioned at all. Nothing about women or their work. But 100% of the time, men and their work were mentioned. And again, that's in a specialty that 42% of the academicians are women. So people read these newsletters all the time, and they think it's fine. Like nobody says anything. Even in my society, in my medical society, when for 4 years in a row, only men got awards, and women were on the stage giving the awards as if kind of like a Price is Right, Vanna White situation, handing out the awards, no one said anything. And we just come to expect, like, wait a second, we can't buy into this. We can't have this established set of attitudes that normalizes this type of behavior that is really reproachable. It cannot be allowed. So moving away from medical societies, but not totally away, because medical journals are owned by medical societies or they have affiliations with medical societies. So let's take a look at that. This is one of the easiest disparities to fix. It's the only disparity that I can think of, and I've thought about this a lot. It is the only disparity that looks this way. There's a more than 20-year evidence base that shows it's a disparity. It's inexpensive to fix it, really isn't hard, and it's really fast and easy to fix. And so if this disparity holds true, one that we've proven over and over again is a problem, one that's inexpensive, fast, and easy to fix, if this one we don't fix, then we really can't fix anything. And of all the disparities, this one really suggests a lack of a will to change, which is why my colleagues and I have gone after this issue of women on editorial boards. And I've been writing about this and basically saying, look, if there are disparities at journals and women can't get their research published as easily, and it sort of seems like the research isn't as good, and maybe it's their fault, and so on, but we know that there's this documented disparity and we haven't fixed it, that is a huge issue. That is a huge issue for deans and chairs that are recruiting academic faculty who need to publish to get promoted. So again, I've been writing about this, and in the writing that I do, I tackle a lot of the arguments that people have. So one of the arguments that people have is, well, medical societies and journals are separate. There's firewalls. We don't, even though we own this journal, we don't participate in, telling you what to do and so on or whatever. Well, what I looked at, first of all, was the International Committee of Medical Journal Editors, and they've codified owner obligations, which say that owners, meaning the medical societies, have an obligation to ensure that editors act in a manner that is compatible with a position of trust. And I really tackled this idea that medical societies can wash their hands of journals while still owning them or still being affiliated with them and allow them to perpetuate. So gatekeeper number 4, funding agencies. We know that NIH and other funding agencies have been very problematic. One of the things about this study that was very concerning, again, for deans and chairs, especially at a top university, is the disparity was even greater there. So when you think about deans and chairs from top universities who are trying to recruit the most expensive, diverse faculty and investing tons of money in their labs and all of these things, and you think about the return on investment for that, if funding agencies have the most bias against those very same people they're hiring, that is a huge return on investment issue for them. And then finally, I just want to touch on sexual harassment, because where there are a lot of disparities and where people are not treated fairly is really excellent grading ground to perpetuate gender disparities, gender-related harassment rather, gender-related harassment or sexual harassment. And so that's where we see a lot of gender-related harassment and so on in medicine. And here's the thing that's really interesting, and a lot of people don't know this. We're really bad in medicine. I mean, we're really bad. We're a lot worse than almost all other industries, except for the military. And when you think about the level of education and the tools we use, the military uses guns, we use stethoscopes and scalpels and other things. When you think about that and you think, wow, we're almost as bad as the military, and we're worse than graduate school and undergraduate school. Like, how did we get so bad in medicine? Like, how did that happen? And again, Francis Collins has come out and basically said, you know, this is unacceptable. And on behalf of the NIH, he apologized to women and said that they were now going forward going to be part of the solution instead of part of the problem. He acknowledged they were part of the problem. And they've done a number of different things. You know, many people would say that perhaps it hasn't been enough, but at least they've apologized, acknowledged they're part of the problem, and are actively doing things to change that. Whenever I give this talk and I can see people's faces and so on, what I see is a lot of, especially young men, with their faces just really sad. And I realized as I gave this talk, especially to people in training, that everyone was thinking, it was not really understanding the data very well. It was thinking, wow, there's a lot of men who harass women in medicine. There's just a lot of bad men. And what I realized, and I'm married to a criminologist, someone who does research on, crimes, and these are crimes, by the way. What I realized is that people don't understand the serial nature of these harassers. In fact, there's so many good men in medicine. The vast majority of men in medicine are really wonderful men. They're ethical, they're kind, they're good people. They may have unconscious bias, and that's important to understand. But in terms of being a harasser, a predator, that's not who they are. And so what has happened in medicine is that the harassers and predators have been allowed to stay and to continue to be predators. And they have targeted many people. And that has really tainted all of medicine in a horrible way. And so we must stand up to the harassers and the predators and make sure and stop them. And so just summarizing a few things as I close here, this is a cycle that we came up with to just show how we should be examining data and, you know, just really taking it through a process, just like you would for a lot of other processes, to have a strategy. And this is a statement on gender equity that the AAMC just came out with. And they looked at leadership, compensation, research, recognition, et cetera, compensation being, you know, super important, but also these other things being important. These are the 4 areas that AAMC said it wanted to tackle. And AAMC saying really for the first time, this is a huge priority for us. Lots of organizations are doing work on this. I just put this up just to show you that there's a lot of information out there, and that you know, the AAMC is really going to leaders and basically saying, you know, we want chairs and deans to be fixing these problems. This is, you know, we want leaders and institutions to tackle this. We have resources for them, et cetera. And this is very important. And this is my chair. My chair actually does gender equity research with us, and that has really opened his eyes. He is a very good person. And if I show him something that where the data is, you know, looks off or there's a disparity, instead of being defensive, he just says, all right, that's what the data shows. Or he might say, what if you look at it this way or whatever, but he is never, ever defensive about the data. The man is a scientist, and he will look at what the data shows, and he will act and adjust accordingly. And that is really what an ethical person does. And so, you know, in closing, ethical leaders do use science. And thank you so much for having me come today. And I really appreciate Dr. Frank and Dr. Kuhn inviting me and really wanting me to present this information, even when I said, you know, coming from Boston, that Harvard Medical School was very early in announcing that their faculty couldn't travel. And they said, even before other places said that their faculty couldn't travel, they said, we want you here today. So thank you very much for supporting this talk.
Thank you, Dr. Silver. That was very thought-provoking, insightful, and just a really great evaluation of the literature, including your work. I'm seeing some comments here, clap, 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 from the audience. We're soliciting questions from the audience through the chat function on Zoom. And one of the first questions is coming from Dr. Lozapone, Kathy Lozapone. Getting women represented on editorial and other review boards is a great start, but how do we deal with unconscious bias of women against other women? Is there a place for anonymous peer review in this?
Right. So that is one thing that's super important to understand, is that women are conditioned the same way that men are conditioned. And so, when we sort of walk through the halls, for example, and we see only male pictures on the walls, or when, we're at an orientation and we see, men coming up and speaking or whatever, or when the national conversation is about how to get PPE and it's mostly men who are talking. Women are conditioned the same way that men are conditioned, which is to respect what men say and to marginalize what women say. And that's really important to understand. That's not a let's blame the women, and it's not let's blame the men. Let's all stand up and recognize that we're conditioned the same way, and we need to reevaluate our own biases and so on. And so, yes, I think it's very important for evaluations to take place anonymous. I think that a lot of journals have gone to anonymous, and even for grants, there's a Canadian study that looked at for grants, if you only look at the work and not the worker, what does that look like? And that took out a lot of the bias. Women got a lot more money for the grants once people didn't know their names. And women often joke, I'm going to change my name to James, or I'm going to change my name to Dave, that'd be really good. Or why did I name my daughters like Emily and Anna? I can't believe I did that. Why not choose something that was more masculine? So.
No, absolutely. Another question coming in from one of our faculty, are these studies, and I actually have a question along this line too. Are these studies with a breakdown between representation for teaching versus research for PhDs versus clinicians, men versus women? What about data tools addressing the intersections? And kind of my question along those lines is, I think, how broad were these categories in these studies looking at the disparities? Because, a PhD woman may be very, very different if they're doing clinical epidemiological research versus a very basic science research.
Right. Yeah, so these are good questions. So it gets to a lot of stuff that has to do with how you do gender equity research. And keep in mind that you can only do research on disparities with the data that you have. So for example, if we say medical societies, we do not know how many physicians in medical societies have disabilities. We do not know how many physicians in medical societies are on the, identify as LGBTQ. We don't know. We can ask them. We don't know. We do not know, in general, how many come from underrepresented racial or ethnic backgrounds because we haven't asked them. Many societies, though not all, have asked gender, so we have that data. AAMC is a good example. They have some ethnic and racial data, but they don't have data on the intersection of gender. And so intersectionality is when you identify with more than one underrepresented group. So you might be a Black gay woman, or something like that. And so, and they tend to the different identities tend to add up to more bias. So we don't have it on gender spectrum, we don't have it on disability, we don't have it on intersectionality and things like that. And so you have to kind of separate out what you can do with research and actual data that you do have and the conclusions you can reach, and how you might apply that to other populations, and also the call for the need to have people identify more and understand, but also recognizing, for example, there may be limitations. Physicians may not want to identify as having a disability, for example.
Great. And I just want to give credit to that question to Tanya Rice. A question coming in from Janice Kerr. Simply brilliant, thanks. Regarding the rare outlier of radiology pay discrepancy in favor of women, is it understood what potential factors may explain this?
So that was a study that was done in U.S. public medical schools, and that may not hold true, for private practice. It may not hold true to private institutions, academic institutions, and so on. So it's really important to understand that that was in one specific population. And no, the causality. So that gets at causality, and that's super interesting because we often identify disparities and then people jump to conclusions about causality, and causality often starts with women should. So that's where, we show disparity in speakers or in elections or in award recognition awards. And then people say women should work harder, women should publish more, women should do this, So causality has not been studied near as much as the disparities themselves. But we have dug deeper into causality sometimes. For example, in the nomination study that I showed, where we showed that no women were nominated who were mid-career or more senior, and the causality is a nomination issue. And then we showed in the committee that women were nominated in the early career, but they never made it a committee. And so part of the solution for that, for what we did, was we had that entire committee undergo implicit bias training. And we did some other things with them as well to make sure that that committee understood that in the past, it had been presented with qualified women and it had never given them an award.
I have two more questions. We'll do them one at a time. One is coming from, and I apologize, I'm not sure who this is. The screen name is Queenie. I will echo the questions about women bias in journals to ask how we approach women biased against women in terms of promotion and career support from female leaders, even physician female leaders.
Right. So these are good conversations to have. I think that there's a lot of people who are, think that sort of women get in their own way because they don't support other women. And I think, and I understand where that comes from. I do understand where that comes from. But I think that the solution isn't to sort of, for women to be mad at other women, but for everyone to understand that we're all trained the same way, which is to not value women as much, and that we all have an obligation. And that's even worse if the woman is a person of color, or the woman, is identifies as LGBTQ, or whatever. So, to just recognize that we all have bias. I have bias. I do this work all the time, and I have, a lot of bias. We all do. And so to check that bias against the data, and to also put in some things that you do routinely to kind of think through, am I being fair? Looking at the paper, looking at the people that you've invited on your papers to work with you, all kinds of different ways that you sort of have checks and balances to look at your own data and how you behave and so on. But I think a talk like this and these conversations are really helpful in bringing up these issues. I think that the last thing I would ever want is women to be even angrier at women, because women already have so many problems and so many barriers and so many things. Like, I don't think that's helpful.
Okay, and that the question asker wrote in and said that was Dr. Victoria Seligman. And so our final question comes for our Vice Chair of Diversity and Inclusion for the Department of Medicine, Dr. Sonia Flores. How do you deal with leaders, mostly male, who claim they are not biased and have advanced gender equity, but still continue to perpetuate the biases?
Right, so that's something, first of all, there's not one easy answer for that. And that can be very treacherous. It can be very treacherous for a diversity leader who is an underrepresented minority to try and tackle. And that's where you really need allies. That's where, men who are leaders, who are actually real allies, can be so instrumental. That's where data helps. You know, when you just keep bringing up the data. Now, some people aren't going to respond to data, or they're going to, if you bring things forward, it could be treacherous for you. And so I'm always cautious about that. I can just tell you, you know, with my chair and the president of my hospital, I have shown them data multiple times, and they will sit down and look at it and listen to me, and it will not result in a treacherous situation for me. But that is not true in, you know, with other people and in other institutions. And you always have to be careful. You really do. You have to be careful, because there are people who will not believe the data and will shoot the messenger, or fire the messenger, or make the messenger leave.
Well, thank you very much, Dr. Silver. I greatly enjoyed your talk today. I can tell from the comments and responses from people coming in through the chat that equally our audience did as well. I wish you health and that the Boston area stays well. Coming in from other people in the audience, and thank you as well.
Thank you, and safe passage to all of you, and thank you so much for what you're doing during this very difficult time. And you know, I hope that the main takeaway is that you will all, you know, just check your own biases, but also just be really kind, you know, especially during this time. Just be kind to as many people as you possibly can. Thank you.
Thanks. All right, I'm going to let Joe, I'm going to put up our award winners, and I'll let you take the mic.
Thank you. So in closing, we'll wrap up quickly here. We'd like to recognize all the excellent investigators and teams who submitted abstracts for today's event. We received 38 abstract submissions overall. The abstract review committee consisted of the 10 current outstanding early career scholars in the Department of Medicine. We were certainly very impressed by all the abstracts and encourage you to review the abstracts online at the ResearchDay website or using the links in the ResearchDay program. We would also like to specifically recognize the two most highly rated abstracts in each of the 5 categories shown here. So congratulations to you all. The winners will receive a small cash prize that we'll follow up with each of you on after the event. And then to close, thank you all for joining this 8th Annual Department of Medicine Research Day. Thanks to Dr. Julie Silver for joining us and for a terrific presentation. Congratulations to Dr. Gutierrez-Hartmann and to our invited speakers, Drs. Rice, Gavin, and Peleya. Thank you to Dr. David Schwartz for your support of this annual event. And finally, a special thanks to Jenny Kemp and Natalie Deliri, whose hard work ensured that we could still gather for this event under challenging circumstances. Thank you all for attending. Take care, and we'll look forward to an in-person event for the 9th annual event next year. Thanks, everybody.